Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
Invisible to the naked eye, they account for the majority of Earth's biomass. For at least 3.5 billion years, they have dominated the evolution of life. So diverse in form and distribution, they are Earth's most ubiquitous life forms, carving out niches in ecosystems ranging from the vastness of the open sea to the digestive tract of a termite. And yet, only 1% of them are known to science. They are the microbes, and without them, life as we experience it would soon come to an end. In fact, it would never have begun at all. We are not alone. You can't see them. You can't hear them. You can't even feel them. But no matter what you do, no matter where you go, they are with you. Tiny, intimate strangers. The microbes. The average human being is composed of 10 trillion cells. An impressive figure, yes? Well, ponder this. For every cell in your body, you harbor 10 times as many microbes. That's 100 trillion microscopic tenants on and in your body. And if the thought of 100 trillion microbes inhabiting your body is making you squirm, don't worry. Without microbes, you wouldn't last a week. From a microbe's perspective, we're just walking, talking ecosystems. For as long as human beings have been around, we and the microbes have been evolving inextricably together. I'm Jim Leinfelder. I'll serve as your guide over the course of this 12-part series, during which you'll be introduced to a category of life that is still largely unexplored. And you'll meet the scientific pioneers who are exploring it. You've all heard the expression, seeing is believing. That's certainly the case in microbiology. True awareness of the widespread distribution of microorganisms and some of their characteristics didn't occur until the development of the very first microscopes. Microbiology, as the name implies, quite literally focuses on organisms too small to study with the unaided eye. They are protozoa, algae, fungi, bacteria, and with the invention of the electron microscope, viruses. But how small is small? This is the tip of an ordinary straight pin. Under an electron microscope, we see it first magnified 30 times. But then, as each image increases in magnification by a factor of 5, we can see increasingly close-up populations of rod-shaped bacteria clinging to the scored sides of the pin's point. And viruses are many times smaller than this. Though the organisms microbiologists study are extremely small, the scope of microbiology is vast, touching virtually every aspect of human existence. In agriculture, microorganisms help break down waste and aid in the fixation of vital nitrogen in the soil that would otherwise come from chemical fertilizers. Microbes are used to treat sewage, clean up industrial waste, and decompose the garbage in landfills. We use them to make food such as cheese and bread. And yet, we do our best to eradicate them from our kitchens. There are bacteria in our large intestine that synthesize vitamins, allowing them to be absorbed into the bloodstream. And herbivores, who depend on cellulose as their major food source, can't digest it on their own. Only the bacteria in their stomach have the enzymes needed to break it down. Cows, sheep, and other herbivores get their energy from the byproduct of microbes. A very tiny minority of microbes act as pathogens or disease-causing agents, representing a natural threat not only to humans, but to all life forms. We therefore attempt to control potentially harmful microorganisms to prevent infection, especially in environments such as hospitals. But it wasn't so long ago that we lacked even the basic understanding of how to control microbes before contamination and infection got a foothold. The results of our ignorance were often dire. Most of recorded history is dictated by disease. During the American Civil War, for example, more soldiers died of infection than of any other cause. Thousands were wounded, allowing microbes into the body, resulting in infectious disease. Because effective control measures were unknown, doctors had few ways to prevent or treat these infections. The microbes took over, the body couldn't defend itself, and the person often died. Delivering mothers of the time faced the same threat. Deaths from infectious diseases acquired during or soon after childbirth were as high as 25%. As the relationship between microbes and disease became clear, control methods were developed, such as the concept of personal hygiene, washing with clean water and soap, and improved public health measures such as a safe water supply and sanitation. Disinfectants, antiseptics, and most importantly, vaccines were developed, and soon we were increasingly effective at preventing infection and disease. However, once infection occurred, we still lacked life-saving treatment. In the early 20th century, doctors could do little more than prescribe isolation and rest for people suffering with diseases such as tuberculosis and many others. There were no cures. Then, in 1928, British chemist Alexander Fleming made a startling discovery. Fleming noticed that a mold he was growing in his laboratory killed encroaching bacteria. The mold was Penicillium chrysogenum, and it produces the antibiotic that we call penicillin. Although we credit Fleming with the discovery of antibiotics, the fact is microbes invented them billions of years ago.
Industrial monument to the miracle drug. Mass production penicillin plant at Terre Haute, Indiana, one of many where the life-saving medicine is now being manufactured wholesale. Tons of mold like that you've seen on spoiled foods are processed by rapid fermentation.
In the 1940s, penicillin became available to the general public. Diseases that had claimed hundreds of thousands of lives for centuries, from pneumonia to syphilis, could now be cured. Today, we can prevent and treat diseases more successfully than at any other time in human history. We still, however, have much to learn about the relationship between microorganisms and humans. Tragically, infectious diseases still devastate human populations worldwide, most often in places where sanitary public health standards are not or cannot be practiced, and appropriate medical care is not available. And there has been an explosion in the number of bacteria resistant to antibiotics, and an alarming but inevitable emergence of new life-threatening microorganisms. But while this lethal minority get most of the media attention, without their less celebrated legions of beneficial relatives, the threat of disease would be a moot point, because life itself would be impossible. It was the microbes that made this planet a viable place for all other life forms. Although plants are generally credited with developing the ability to convert the sun's rays to food energy, photosynthesis, it was actually ancient microorganisms, the cyanobacteria, that first produced oxygen as a byproduct of photosynthesis 1 billion years before the first plants ever existed. Over the next 3.5 billion years, microbes evolved to fill every niche, creating an environment in which plants, animals, and humans thrive. Microorganisms now maintain balance in the environment by playing a key role in the recycling of vital nutrients such as nitrogen, sulfur, phosphorus, carbon, and oxygen. Today, all over the world, plants depend on microorganisms to feed them nutrients. In turn, the plants supply 50% of the oxygen we breathe. The rest comes from marine microbes, especially algae and cyanobacteria. A visible form of algae is seaweed. Like plants, algae are photosynthetic and help to anchor the food web in the ocean. Marine bacteria and algae help maintain the world's supply of oxygen by playing an essential role in the carbon cycle. Primary producers, such as algae and plants, draw energy from sunlight, taking in carbon dioxide and releasing oxygen. Consumers, such as animals and many microorganisms, use oxygen and give off carbon dioxide. It's an endless cycle, and only one of the countless ways microorganisms have supported life throughout our planet's history. Without this cycling of nutrients, the surface of the Earth would be as barren as the moon. The invisible microbial world is rich in its diversity. It is populated by more different kinds of organisms than all of the visible world combined. The diversity of all life is derived from the microscopic crucible of life, starting with the first microbes. Microbes reproduce quickly. Their generations fly by. And because they've been around for billions of years, they've had the opportunity to shuffle and reshuffle every possible combination of their genetic information, their DNA. As the environment changes, so do the organisms, which in turn can change the environment. As scientists have begun studying DNA, a molecule shared by virtually every living creature, they've discovered a method of comparing the ancient pedigrees of organisms to look back in time and unlock the mystery of how all life is related. This technology is also being used in a myriad of ways, from mapping the human genome to developing new antibiotics. But microbiology is still a very young science. We actually know very little about most of the Earth's microbial tenants. Scientists across the globe are searching for these unknown microorganisms and their role in the subsistence of life. The first step in this search is their discovery, collection, and identification, a step we have barely begun to take. Marine microbiologist Steve Giovannoni is combing the Sargasso Sea for microbial life that will help us understand how life works in the world's oceans. A generation ago, scientists failed to appreciate the vast diversity of marine microbes.
There were plenty of skeptics. There were people that thought that the kinds of bacteria that we already knew about were a perfectly good example of what lived in the oceans.
Giovannoni and his team have returned to Oregon State University to look for undiscovered microbes among the samples of water from the Sargasso Sea. But how do you know when you've found a new microbe anyway? Searching with a microscope is not realistic because so many different types look the same. Imagine this is Giovannoni's mystery microbe, unrecognized in a sample of the Sargasso water. When he looks, he can't identify his quarry simply by physical appearance.
We had to find some other way of identifying them. And what we did was to become gene hunters. In place of the organisms, we began to hunt their DNA.
Giovannoni chemically broke open millions of different microbes. The messy results were billions of different DNA fragments, all mixed together. DNA is a complex genetic recipe that consists of two strands held together by four chemicals called nucleotides, which are like puzzle pieces. The length and arrangement of these four chemicals determines the identity of every living thing on Earth.
We removed the DNA from the microorganisms in the seawater, and we took single genes. We took single copies of genes from all that DNA, and we studied those single genes. And we did it by reading the code on the gene.
Giovannoni retrieved pieces of a specific gene, the one coding for ribosomal RNA. Scientists used this gene to identify organisms. He compared the ribosomal RNA with an up-to-date database of existing codes, the genetic codes of all known living things. One by one, the codes matched existing organisms. But one code did not match up. It belonged to an unknown organism. They had discovered the existence of the mystery microbe. They named it SAR11, Sargasso gene number 11. Now the real work would begin. He had to find the whole microbe. It wasn't long before the search took on greater significance. Giovannoni's research team found SAR11 in water everywhere they looked. And they knew it was a bacterioplankton, which means it is involved in some way with the supply of nutrients to algae, the oxygen producers of the sea. Following Giovannoni's lead, other scientists found the same gene in ocean samples from around the world. Evidence even turned up in freshwater lakes. Even more remarkable, in every sample of water, there were more SAR11 genes than genes from any other organism. Giovannoni realized that he was on the trail of possibly one of the most abundant life forms on the planet.
Right now, all we know about SAR11 is one gene, and we know that gene is out there in the sea.
Stephanie Conan is a graduate student in Giovannoni's laboratory.
We feel that it plays an important role in the food web in the ocean, and we would like to find out what that role is.
But so far, they hadn't managed to grow SAR-11. And for over a century, the only way to study microbes was to grow them.
This is one of the oldest methods in microbiology. It's called a spread plate. And really all it is dropping water from the environment on the surface of a sterile plate that contains food for bacteria, separating the bacteria so that they grow up by themselves and form colonies that we can see with our eyes.
1 week later, so many bacteria have grown that they become visible. For years, microbiologists believed that the colonies which grew like this on spread plates revealed the majority of the microbial species on the planet. The reality was different. Over 99% of all microbes on Earth will not grow this way.
The real challenge today is to try to grow SAR-11. Something about these cells makes them unable to grow in a scientific laboratory, at least today, using the kinds of techniques that scientists have. I told Stephanie this, and I said, what would be really a great discovery, but challenging and require creative thought, would be to try to isolate these organisms for the first time. Do you want to do that project? And she said, yes.
We are going to be testing different food sources for this organism to determine what it eats. And if we can do that, we can start determining what its role is in the food web.
Our hypothesis is that SAR-11 eats organic carbon, but what we don't know is what kind of organic carbon does it eat? What is it like to eat? And that's one of the things Stephanie has to figure out in order to make it grow.
If Stephanie's diet for SAR-11 does get it to eat, grow, and reproduce, they will be able to find out much more about what the microbe actually does. Unlike many microbes, SAR-11 thrives with very little nutrients, so Stephanie is tempting it with very small amounts of food, 1,000 times less than normal. But it's a stubborn microbe.
We think that the reason it's so difficult to grow in the laboratory is that possibly SAR-11 is a small cell built to live at the very low nutrient concentrations that are found in the open ocean sites that we study, and that it's precisely adapted to do one thing, and if we change anything, then it doesn't divide in the lab. So the trick for us will be to somehow or another reproduce exactly what's happening out in the ocean in order to make it grow.
Although she has not yet been able to get SAR-11 to grow, Stephanie's persistence paid off in a different way. She used SAR-11's single gene to get the mystery microbe to reveal itself and pose for a photograph.
Under the best microscopes, SAR-11 is nothing more than a pinpoint of light. It's like looking up at night and seeing a star in the sky. And they're a lot smaller than most of the other bacteria out there. In many cases, they're 1,000 times smaller. You could put 1,000,000 billion of them into a tablespoon. What's it doing out there?
What is SAR-11 doing out there? That's a question that applies to the vast majority of microbes on the planet. We know they have great power, playing paramount roles in disease and its treatment, in industry, public health, and in the Earth's evolution and very functioning. But we know so little about so many microbes. To know more, scientists are studying the molecule from which all the functions and diversity of life is derived, DNA. In the next episode, we'll explore how DNA found in all living things is expressed in the same fundamental structures of all life. You probably never thought you had much in common with microbes such as Salmonella, Staph, or E. coli. But the truth is, you do. All living things, birds, fish, worms, lizards, grass, you, me, and all the microbes thriving underwater, underground, in the air, and inside our bodies, we're pretty much the same at the molecular and genetic levels.
Well, they're putting up a new building here. Not exactly sure what it's going to look like. Sure hope the architect knows. But what I can tell you is the fundamental concepts are the same on all construction sites. Raw materials are put together by builders to create new facades and infrastructures. And it's the same with living cells.
Whether they're algae, bacteria, fungi, plants, or animals, their raw materials, such as proteins and nucleic acids, are similar, and they combine together in ways which realize a host of possibilities.
I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll explore not only the vast differences between living cells, but the cellular functions and structures that all living things have in common.
Those cells can look amazingly different. Each one faces the same challenge of getting energy and reproducing. Every cell needs nutrients and an environment specifically suited for its metabolic needs in order to carry out these functions and survive.
While a cell's internal architecture turns out to be different depending on whether it's a prokaryote or a eukaryote, all cell processes and products are similar and often identical.
Eukaryotes, like the cells of plants and animals, are designed like this office building with many floors and rooms. Eukaryotes are complex and compartmentalized, including structures such as lysosomes, mitochondria, ribosomes, and chloroplasts. They have an internal membrane structure and a true nucleus to enclose their DNA. Cell structures may differ slightly, but plants, fungi, and animals, including you and me, share the eukaryotic cell plan. Bacteria are prokaryotic. Prokaryotes are designed kind of like this baseball stadium. The floor plan has very few internal structures. Most prokaryotes have a cell wall. Some have a slime layer around the outside, but none have a distinct membrane-enclosed nucleus. The DNA is found free-floating, along with ribosomes responsible for protein synthesis. Several of the functions found in the membrane-bound organelles of eukaryotes, like mitochondria and chloroplasts, are also found in the cell membranes of prokaryotes. Many bacteria can move around using appendages called flagella. Membranes allow food in and waste out. They have all the necessary material and machinery needed to grow and reproduce. Proteins embedded in the cell membrane initiate duplication, following the code provided by the DNA strands inside. The cell then divides into two complete bacteria.
But even though prokaryotic cells can be as different in form and function from eukaryotic cells as an office building is from a baseball stadium, let's not forget, they do share common traits. They both have cell membranes and ribosomes and contain DNA as their genetic material.
In fact, the mitochondria and chloroplasts present in most eukaryotic cells are similar in dimension to prokaryotic cells. Coincidence? Not really. At some point in evolution, free-living prokaryotes took up residence inside primitive eukaryotic cells, becoming the first internal structures, or organelles. This process is called endosymbiosis. An abundance of microorganisms are hosted by the human body in areas such as the upper respiratory system, on the skin, and in the digestive system. The large intestine has hundreds of species of microbes living in communities. They exist cooperatively, digesting food and making vitamins.
Hi, thanks. Okay, got your eukaryotes like us. You've got prokaryotes like bacteria. They're all cells, they've all got membranes, DNA, RNA, but just when you think you've got it all figured out, nature throws you a curveball.
The common cold, influenza, chickenpox, and measles. You've heard of them. In fact, you might be carrying around the organism that causes one of these illnesses right now. But unless it's making you sick, you might not even know it's there.
Viruses. These tiny, I mean tiny, well, let me give you an example. Let's say the virus is the size of this baseball. Well, then a prokaryotic bacterium would have to be something considerably larger. Maybe the size of the pitcher's mound. And a eukaryotic animal cell would be about the size of the entire infield. Oh, right. Anyway, these tiniest of troublemakers may seem like they function like other organisms. But the truth is, they're not even considered to be cells. In their basic form, they're as inanimate as, well, this hot dog.
It takes contact between a living cell and a virus to bring it to life.
In fact, a virus is like a baseball in other ways.
Here you go.
Thanks. Hey, nice job. If you examine a baseball by, say, oh, I don't know, cutting it in half, you begin to get a sense of how some viruses are organized. The outermost layer of the virus, in this example, the horsehide on the baseball, is called the envelope, which is complex and can vary chemically. Some viruses don't have an envelope. This next layer of tightly woven material here, that represents the capsid or protein coat, something which all viruses do have. And then this innermost rubber core, that represents the part of the virus that contains its genetic material. I'll take it from here, Bob, thanks. A virus can't replicate on its own. So because it lacks its own manufacturing plant, it has to invade a cell and use the host cell's materials and equipment to do the manufacturing. But what does this have to do with pizza, you're asking?
Let's say that this pizza is a virus and it wants to replicate itself. It would have to invade this pizza parlor, which has all the ingredients and equipment, including me, that it needs to get the task done, just as a host cell has everything a virus needs to reproduce. A virus is the smallest and simplest of microbes. Unlike a bacterium, it doesn't have the ability to reproduce on its own. It must enter a living cell. Once inside, the virus sheds its outer protein coat. It hijacks the host cell's machinery in order to reproduce. The long chains of viral genes are duplicated by viral proteins made in the host cell. Once all the parts for the new viruses are completed, they reassemble. Within hours, a single infected cell can give rise to 1 million new viruses. They destroy the host cell and go on to infect new cells. The virus and cell interaction can sometimes cause disease. Humans aren't the only ones who can carry these tiny microbes. It turns out that all living cells can be invaded by viruses, from plants to pets and even bacteria. Viruses may seem like they don't have much in common with prokaryotes and eukaryotes, but actually, their chemical needs are quite similar. Although viruses all do the job differently, by borrowing a host cell's machinery, they all successfully achieve their goal of reproduction. And if this pizza were an actual virus, this parlor would be floor to ceiling in pepperoni and cheese. All forms of life, prokaryotic, eukaryotic, viral, even construction workers, may seem different, and in many ways they are. But most of their central processes are the same. They must all use energy as ATP, along with building materials to reproduce. Enjoy it, gang. Baked it myself.
Oh, this looks good. All right. Hey, thanks, man.
They also have to store genetic information. Without blueprints, these construction workers wouldn't know what to build. The same goes for cells, only the blueprint, or genetic information, is in the form of DNA, or in some viruses, RNA. With a different blueprint, you get a different building. And with different nucleic acids, you get different organisms. The metabolic functions that all organisms have in common are the real-world expressions of their DNA. They're like the construction workers who take the building materials and put them together according to the blueprint to make the structure we see. Scientists have discovered that all living things share parts of the same blueprint, shown by the common features present in all organisms. What we understand about our own species comes not only from studying our DNA, but also from studying the DNA of the microbes that evolved before us. Human beings haven't been around long enough to invent hardly anything new. We are largely built from the inventions of other species, including microbes. They came up with the inventions like shutting down for a few hours every day, which we inherited as sleep, as well as the basic mechanism that makes it possible for us to see. But even then, so what? One way or another, we have become marvelously capable, highly successful beings. And why on earth should you care about how it happened? To find out why you should care, take a trip with one of the people who is helping to decode your DNA. Meet Dr. Craig Venter. A consummate sailor, Venter and his crew are on their most ambitious journey ever. A voyage along the path that Columbus sailed from Spain to the Caribbean and the New World.
Venter has been responsible for key breakthroughs in today's genetic revolution. After establishing whole genome sequences for many bacteria, his latest project is the sequencing of the entire human genome. Venter attributes his accomplishments to his different way of looking at the world.
I think each of the breakthroughs that I've been lucky to come up with my team have been because I go outside the box. It's much easier to go outside the box than trying to work up linear through a maze. You know, you go outside and you go around the traffic instead of trying to go through it all. And I think just by doing that, I've been able to get places and see relationships that other people don't see.
Some of the genes that make up the human genome have already been decoded by Venter and others. And perhaps the most remarkable things that research reveals is how much we can learn about the inner workings of your body from studying the inner workings of microbes.
It's clear every gene in the human body can be traced to genes in other species. We have 60 to 80,000 human genes. A bacteria has 2,000 genes, but we can trace back origins of all of our genes to things that we find in the archaea and in the bacteria.
Knowing the entire gene sequence of a microbe gives us the ability to compare our genes to other organisms. This has given rise to new insights into what happens when things go wrong inside us. Take colon cancer, for instance. It turns out that many people who get colon cancer are missing a gene. How did we discover that?
Those genes were discovered in terms of their function by aligning the sequences in the computer that we have obtained from human cells to those from the bacteria that lives in our guts.
Scientists found a gene in these bacteria which codes for an enzyme that repairs damaged DNA, a precursor to mutation. Then, Venter and a colleague compared the bacterial genome with a part of the human genome. They discovered the exact same gene in human beings and found that it helps keep us from getting cancer, also by repairing damaged DNA. People who are missing this gene can now learn that they are at risk long before cancer develops. Knowledge that will save tens of thousands of lives. What microbes are ultimately telling us is that each of us contains within our genetic material a written history of life on Earth. Take, for example, these human cells. From studying the DNA inside them, we have learned that cells like ours were born over 1 billion years ago by a marriage between different single-celled microbes.
Our basic cells are a combination of multiple organisms. We're composites of bacteria, lineages from bacteria and other microorganisms. So it's compounding life on top of life. Each genome is the recorded history of life.
After 15 days at sea, Venter and his crew arrive at their ultimate destination, the island of Antigua. In June of the year 2001, Venter expects to arrive at another destination, the completion of the sequence of the human genome. We stand on the verge of an unequaled explosion of knowledge about who we are and how we came to be here.
The ultimate point of most science is to try and understand who we are, where we came from, how we fit into the environment, how we fit into the overall picture. Did we extend out from life of these simple bacteria? Did we come from outer space? You know, who are we? What are we? You go out of this context of the laboratory and you go out and look at a tree or a fish or, you know, things I encounter on the beach, and I look at them and I realize that we share, you know, 90% or more of our genes with those other species.
Our new understanding of DNA has shown us that, beyond any doubt, we are beings whose roots lie in the 4 billion years of life on this planet.
Over 4 billion years ago, we didn't have intact cells. Life was a series of chemical reactions on the surfaces of metals, other things in the ocean, to eventually develop a cell that became self-replicating with the duplication of the DNA molecule. That eventually, over billions of years, became us, became all the other living species that are present on the Earth today.
But knowing this sometimes makes us uneasy, because ever since the first time we told a story about our origins, most of our stories have portrayed us as beings apart.
I find it empowering. I don't find it a threat to my humanity. I think it helps define humanity. With genetics, it's the intimate, minor differences that are everything.
In the coming years, we are certain to learn far more about the ways in which we're linked to the rest of life on Earth, and the ways in which we're unique. But no matter what lies ahead, it is unlikely that we human beings will ever again be able to view ourselves as a species apart. From one point of view, that may seem to diminish us. But from another point of view, we are gaining a deeper understanding of our role in what may be the most miraculous story in the universe. Life on Earth. And that new understanding can only serve to increase our sense of wonder. From the largest organisms to those that are rooted in the ground, to those that fly, swim, float, dig, to those too small for the human eye to see. All living things have common metabolic and reproductive processes. They store information and energy in the identical types of chemicals. We are all connected.
In the next episode, we'll explore those chemical reactions and see how the similar needs of all organisms manifest themselves as differences in metabolism.
As different as all organisms are, multicellular or single-celled, they all share similar chemical molecules like DNA. It is the DNA which determines how each performs a fundamental process, making and using energy. In other words, how they metabolize.
How do organisms survive in this fickle and competitive universe? Well, for one thing, We all need food. Chemicals we can break down and use as energy to build new cells. This apple works fine for me, but it wouldn't satisfy a hungry carnivore. Different organisms, different energy needs.
No matter what the habitat a microorganism may call home, all microbes are performing this one basic life process. A process we all must perform to survive and grow. Metabolism. Metabolism refers to the thousands of chemical reactions and physical workings of the cell. It is the flow of matter and energy.
I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll explore a variety of energy-producing metabolic strategies. Strategies determined by the molecules shared by all organisms, DNA, the backbone of diversity, protein synthesis, metabolism, and life itself.
Cells are like chemical factories. They spend a lot of their time producing energy as ATP, adenosine triphosphate, the energy storage material of all life on Earth. They then use that energy to drive their chemical reactions. These chemical reactions are divided into two groups, catabolism and anabolism. Catabolism is the process of breaking down macromolecules into their smaller component parts. These components may either be further metabolized for energy or used as building blocks for making new macromolecules. This process of using the energy and simple building blocks to make new macromolecules for the cell is called anabolism. It is an organism's environment and the energy sources available in that environment that determine which energy-producing strategy is successful, and therefore which organism will survive. Organisms can obtain their energy in one of several ways, such as photosynthesis, the conversion of light energy into ATP, or chemolithotrophy, converting inorganic chemical energy, such as hydrogen gas or sulfur, into ATP, or fermentation and respiration, the conversion of organic chemical energy, such as organic waste or sugars, into ATP. They are different processes, but all can go on in the same environment, and some even in the same organism, at different times or under different circumstances. The way that life works is that primary producers, such as photosynthetic and chemolithotrophic organisms, use part of the ATP they produce to create carbon-rich compounds. Other organisms, consumers, break apart the bonds in these carbon-rich compounds to release the energy that has been stored. Primary producers, such as plants, form carbon-rich bonds, and consumers, such as animals and microorganisms, break them. It is one big cycle. Microbes, since they have been here longer, have evolved more ways to do it. In most of the world outside the laboratory, ecosystems are complex mixtures of organisms with different metabolic abilities that not only coexist, but function together to be more than the sum of the parts. Like this ecosystem, a wastewater treatment plant in Radford, Virginia. Despite all the hardware, it's metabolism-driven. Waste materials coming into a wastewater treatment plant come from a vast array of people who eat different things and have different microbes and therefore excrete different things, as well as anything else that goes down the sink or toilet.
Throughout this plant, there's a frenzy of metabolic activity. But we're really here to focus on one, respiration. And to help me with that is Dr. Nancy Love, a professor of civil engineering at nearby Virginia Tech. And you're really here to talk
Activated sludge. I love to talk about activated sludge.
Hey, who doesn't?
But before we talk about that, that's further down in the process. We need to spend a minute talking about this.
Okay, what goes on here in this big pond, basically.
Well, the wastewater that comes in from the community, as well as some industrial inputs, comes in here, and you have to separate the liquid from the solids. And at this point, all we're doing is removing large solid particles from the liquid. And then the liquid is what we're going to pass on down to the activated sludge process, where the metabolism is occurring.
Oh, this isn't where the microbial action is.
Exactly. But do you want to go see that?
Yeah, let's get to the metabolism. Dr. Love, when my layman's ears hears the word respiration, I immediately start thinking about breathing, but that's not exactly what we're talking about here.
That's right. I mean, the organisms are breathing in a sense, but it's more complicated than that. The wastewater that comes into this plant has energy in it in the form of electrons, and the bacteria convert those electrons into a form they can use. It's a food source for them, it's an energy source. These compounds in the wastewater enter into the cells, the bacterial cells themselves, and when that happens, the available electrons have to go somewhere. And in this case, those electrons go to oxygen. So the organisms park the electron from the food source, the wastewater, on oxygen, and in the process of doing that, create ATP. That process of taking the electron from the wastewater compounds, parking it on oxygen, and producing ATP for energy, that process is respiration, and it is a form of catabolism. At the same time, not all of the compounds in the wastewater go through catabolism. Some of them go through another pathway and create body parts or cell materials to form new cells. So what happens is you have wastewater coming in with available energy, and it ends up forming water from the respiration process on the catabolic side and cells on the anabolic side.
And it all has to take place in the presence of oxygen, and the more oxygen, the more readily and rapidly it will take place.
Absolutely. There's a lot more energy coming into the system than the atmosphere has oxygen to satisfy. So it's very important to provide a lot of air, and that's what you see here, the swirling in the basin and swirling through the reactor, so there's plenty of oxygen available for all the organisms.
The mixture of wastewater and bacteria, which engineers call mixed liquor, is then brought to this clarifier, where the millions of bacteria and other particles settle.
The liquid goes through a few more processes and then into the river, but the solids go to one of two directions. Most of the solids, these bacteria that we created before, go back to the front of the aeration basin so that we can start the process of metabolism all over again. But if you think about what we talked about up there, the wastewater coming in enables the growth of more bacteria. And if we don't waste or remove some of those bacteria, we'd be up to our ears in bacteria in no time. So it's very important to remove as many bacteria as you make in one day. We waste what we make. So another fraction of the solids at the bottom of this clarifier are taken off to another side of the plant where we handle the solids. That's called the waste.
The solids to be wasted are transferred to an anaerobic digester. Within the anaerobic digester, a diverse community of bacteria perform complex fermentation reactions, which further reduce the volume of the waste, producing organic acids in the process. Other bacteria, called methanogens, convert the organic acids to methane gas. So, the anaerobic digester reduces the volume of the waste by converting solids to gases, enough gas to lift the tank's 125-ton lid.
So, Dr. Love, I take it we've finally reached the end stage of this entire process here.
That's right. What you have here are the residual bacterial cells. Remember, back up at the earlier part of the bioreactor, we had solids that we said we had to waste. We waste what we make. And those solids went through several different processes where we removed water, we anaerobically digested the solids, where we killed the bacteria and we killed any pathogens that were present in the solid material. We removed some more water, and then this is what you have.
Essentially what we've got here are the corpses of all those bacteria.
That's right. They're corpses, but there's a very good nutrient value associated with this material. This material is actually applied to agricultural lands.
Many different nutrients remain in water emerging from a wastewater facility. Primary producers need such minerals to start the carbon fixation process over again. For the most part, this process is driven by the sun's energy, light. But what happens when there is no light? No problem.
In the late 1970s, scientists in an improved deep ocean submersible were able to explore much deeper than ever before. At that time, it was believed that no life existed in the deep ocean. Over 2 miles down, scientists found underwater volcanoes spewing superheated minerals from the Earth's core. When these minerals meet the ice-cold water, they crystallize, creating huge undersea structures. One is 15 stories high. Spotlights pierce the blackness. There is no sunlight. Yet life blooms in the darkness. How? Microbes. Instead of sunlight, bacteria use the sulfur from the volcanic vents as the basis of their energy metabolism. These organisms are called chemolithotrophs. They convert inorganic chemical energy into ATP. The bacteria become food for some of the larger creatures and form the base of the food web. Tube worms, shrimp, fish, and crabs thrive in this oasis of heat and microbes. Much of this is still a mystery, but one thing is conclusive. These chemical-powered bacteria are at the center of this deep-sea food web, and the darkness of the ocean floor is not their only domain. The peaceful emerald forests and dynamic pools of the Pacific Northwest Cascade Mountains, a haven for photosynthesis. Business as usual for the microbes that flourish here. But chemolithotrophs, not entirely unlike those found beneath the surface of the sea, have been present over billions of years, waiting for an instantaneous, cataclysmic change in the environment that will give them their chance to metabolize and grow. In May of 1980, Mount St. Helens erupted, changing the surrounding environment drastically. The balance was tipped, and suddenly sulfur became abundant. The small population of organisms that metabolize sulfur suddenly had the opportunity of a lifetime, the opportunity to take hold and flourish, and they took advantage of it. Environments change, DNA changes, new niches are filled, and metabolism goes on. Some things change slowly, and other things change rapidly. In changed environments, organisms adapt to have a better chance of coping, surviving, and possibly prospering.
Mount St. Helens and ocean sea vents are extreme habitats, at least from the perspective of a human being. But home is home to a microbe, anywhere food can be found. Another type of metabolism is one we human beings have gotten pretty good at using for our own purposes: fermentation.
Baking bread, making cheese, and brewing beer. Very different habitats and systems, but the metabolic process is similar.
People have been enjoying bread, cheese, and beer for thousands of years. However, early bakers, cheesemakers, and brewers really had no idea why yeast made bread rise, or formed holes in Swiss cheese, or turned hops and grains into brew.
What they were doing was tapping the power of the microbial world, harnessing the metabolism of microbes, and enjoying the products of microbial fermentation. Fermentation is one of the oldest metabolic processes on Earth. It is a simple metabolic strategy in which organic compounds are broken down in the absence of oxygen into simpler organic compounds. In the case of bread, beer, and Swiss cheese, different organisms are involved to produce each product. But in each case, the organism uses fermentation. Simple compounds, such as sugars in the grains and lactose in milk, are broken down into simpler organic compounds like alcohol and CO2. The CO2 is what makes the bread rise, the beer fizz, and the holes in Swiss cheese.
And whether it's cheese, bread, or beer, what's happening in addition to fermentation is growth. And as far as the microbes are concerned, growth means reproduction.
No matter how big the vats, the process all happens at the microbial level, one cell at a time. And it has to be the right cell. Jamie Emerson is a brewmaster in Hood River, Oregon. For him and every brewer in the world, brewing starts in the lab.
Jamie Emerson, Jim Leinfelder.
Hi, nice to meet you.
Nice to meet you. What are you looking at in the petri dish there?
Well, this is brewer's yeast. It's our favorite microorganism around here. What we've done here is used a plate and separated out single colonies of yeast, and you can see them as single dots here right on the media. And this media is basically provides growth material for the yeast to grow on, and then we can look at these cells individually.
So those are good results then?
Good results. If you would have seen anything that we didn't want on this plate, it would look blue because the media would give it a dye and colors it.
Okay. And so when you see one yeast isolated there, then you know that you can put these guys to work for you.
Absolutely. We can take a single cell here and begin growing it and actually use it to pitch a large fermentation tank with trillions of cells.
Wow. And here we've got, what, maybe?
Oh, a couple thousand colonies of yeast, give or take. Then with all these plates behind us, we use that to track cleanliness of the brewery, because basically brewing is 99% cleaning and 1% art.
Right.
And we use those back in an aerobic and anaerobic chamber behind you. So this is where we keep track of what's going on in the brewery from an aerobic standpoint on one side and anaerobic on the other. That way we can keep track of both kinds of ferments. Ours in particular are anaerobic. That's what we're mostly interested in.
So essentially you're basically a yeast farmer.
Yes, I am. I'm a yeast farmer, and here we're just checking that we're farming right, that everything is right, very clean. And you know, it takes a lot more than just what's in these petri dishes to make beer. And I'd happy to take you down and show you the farm.
Oh, great. Love to see it.
Well, Jim, welcome to the back 40.
I guess this is where the farm analogy starts to break down, but this is where you're growing more yeast and also starting the fermentation process?
Absolutely. You remember in the lab, we had the plate with just a few cells on it. Yeah. We take a little bit of those cells and add it to the wort, which is unfermented beer. It's full of sugar. The yeast eat the sugar up through metabolism, making energy, more yeast. We keep adding more and more liquid in this tank till it's absolutely full. And in that time, the yeast are chewing up all the sugars, growing more yeast. And we're talking in the range now of trillions of cells in this tank. And then this tank is then pumped into one of the large fermenters, and we fill this fermenter all the way up. Okay. And by the time that process is finished, we're talking 7 quadrillion cells.
Jamie, how do you know that these 7 quadrillion yeast cells are metabolizing the way you want them to metabolize?
Well, besides the information we get from the lab, we actually take it out and look at it. Well, here you can see what's actually going on inside the fermenter. This is beer that's undergoing ferment. The liquid we ran in is absolutely clear, and you can see the haze in there, and that is the yeast cells that are multiplying and chewing up the food in there. You can see the bubbles coming up to the surface, and that's a byproduct of the fermentation. Yeast is metabolizing the sugar and making CO2 and alcohol.
As fermentation continues, the CO2 is vented off to prevent the increased pressure from killing the yeast and prematurely stopping the desired fermentation. The yeast cells continue to grow and grow to reproduce. The growth of yeast cells, as well as bacteria, can be expressed in a growth curve. The number of cells is plotted against a timeline. In the initial or lag phase, the population remains static as the organisms get used to their new environment. Next, in the exponential phase, cell growth increases exponentially, doubling with each generation, until the species reaches its metabolic peak. The peak soon becomes a plateau in the stationary phase, as the rate of growth or reproduction equals the rate of death. Death occurs for a variety of reasons. An essential nutrient may run out, waste could reach toxic levels, or a predator may appear. Whatever the reason, the population then enters its death or decline phase, and the organisms rapidly die off.
Once the yeast are done eating all the sugar, we can take them out of the tank, and here's the granddaddy of them all.
Oh, how do you know when they've finished eating?
Well, we only give them so much sugar, so when the sugar's gone, they're done.
Ah, okay.
Well, this is the culmination of our process here. Collecting some yeast.
Ew, so that's what's coming out of the bottom here?
You got it. And it may look like quite a bit. You know, there's several thousand million yeast cells probably in here.
Yeah, it looks like a malted milkshake.
But it's all one kind of yeast, all genetically identical, which is the trick with fermentation. There's lots of things that ferment, but we're only looking for one thing.
So this is a monoculture.
Monoculture.
I mean, talk about reproduction equaling growth. You started with a vial, and you end up with truckloads of this stuff.
That's right, and you want to see some growth just out of one tank.
Wow.
Gallons and gallons and gallons of microbes.
As the environment changes, new niches arise, and the microbes change to fill them. Over time, the number of niches expands, and so do the different kinds of microbes. As they develop new abilities, some of them keep all or part of the old information in their DNA, and that lets them respond if conditions change again.
As different as these organisms are, and as varied the means of metabolism, all living organisms have the same goals: to make and use energy. The instructions for new generations and all the metabolic activities for all forms of life on planet Earth are passed along through DNA. It is this amazing molecule that we will focus on in the next episode of Unseen Life on Earth. The notes in a chord fit together to make a complicated single sound that works as a whole. Just like the bases of DNA combine to make a complicated whole. But what happens if a chord is off by just one note? Yikes! And what happens when a base is changed in the DNA? Well, it may not happen all the time, and when it does happen, you might not even notice it. But when it does make a difference, things change.
I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we're going to decode the code of life and examine microbial genetics. The source for all biological information, all the information for life, is contained within every cell, inside DNA molecules. Hey, you're sounding good in here, buddy.
DNA, composed of 4 nucleotides, holds the code for making proteins, the components of our cells that perform the work and provide the cell structure. DNA does this in our cells and, in fact, in every other living cell, whether it's a bacterium, a bear, or a begonia.
code. And by itself, it's just a code. Just like these notes are just a code until they're translated into sound. In order for cells to function, the DNA information has to be transcribed and translated.
Transcription, synthesizing messenger RNA from DNA, is like taking the DNA storage file of information and making a copy from it you can use and edit, kind of like recording music. Messenger RNA then takes the DNA information to the ribosomes. Once in contact with the ribosomes, translation occurs, similar to the way this music has been converted to a computer file. Ribosomes read the 3-base codes, and transfer RNA molecules bring the corresponding amino acids. This process continues, and proteins are made. Sometimes changes occur in the DNA sequence. These changes are called mutations, and they can make a big difference. When mutations happen, they produce genetic variety. The more genetic variety, the better a species' chances for survival, which is good if you're a microbe trying to cope in a changing world. In bacteria, the effects of mutation are seen more frequently because generations pass more quickly, and new traits can be passed to many offspring. But there are mechanisms for repair of DNA that have evolved, making mutations rare events. Cells have several enzymes that can detect errors in the DNA sequence. During replication, if the wrong nucleotide is inserted and the nucleotide codes don't zip up just right, enzymes fix it. They recognize errors in the DNA sequence and replace incorrect nucleotides with the right ones. But every once in a while, one will slip by. It's really rare, about one mutation every 100 million times of replication. Most of the time, the mutations that occur present a problem of some sort, and the cells don't survive or pass on the change. Occasionally, a mutation will be beneficial for an organism, as in the case of antibiotic resistance for bacteria. These mutations are maintained, and species may evolve. But what may be beneficial to a microbe can be detrimental to us. This is illustrated by one of the greatest medical challenges of recent times. Vancouver, Canada. Julian Davies is a scientist, teacher, and entrepreneur whose microbial research could save thousands of lives.
And most of the antibiotics that we have at the moment, we've been using for almost 50 years. And in 50 years' time, microbes have learned how to live with them. There are microbes that are found in hospitals that are causing severe infections in human beings that are resistant to every antibiotic that we know. They cannot be treated.
Tuberculosis, long considered under control in the developed world, is returning. So are many other illnesses we thought antibiotics could cure. Even though antibiotics work in different ways, microbes are outsmarting them, one by one. Disease-causing bacteria colonize living tissue. Penicillin kills the bacteria by weakening their cell walls. When the walls collapse, the bacteria die. The patient's disease symptoms subside. Microbes reproduce by dividing in two, replicating themselves exactly each time. But mutations, the random genetic changes in all living things, are seen more frequently in microbes because they reproduce so rapidly, as often as every 12 minutes. When a mutation enables a microbe to fend off an antibiotic attack, all the better for the new, stronger microbe, and all the worse for us. As antibiotics kill off the susceptible microbes, the resistant ones can multiply quickly, without competition. So the more antibiotics we use, the more the playing field is cleared for antibiotic-resistant bacteria.
People ask for antibiotics when they shouldn't need antibiotics. Doctors prescribe antibiotics when they should not. As much to 40% of prescriptions written by family physicians are inappropriate. You don't give antibiotics for a cold.
Antibiotic resistance has reached crisis proportions. Around the world, thousands of people are dying of infections that existing antibiotics can't touch.
We have to remember here that no matter what we do, microbes will become resistant to antibiotics. It is inevitable. What we can aim for all the time is to slow this process down.
Nothing will stop microbes from mutating. It's their nature to change. Actually, real mutations generally don't help organisms conquer Earth overnight. But the DNA code can be misread at times, and small changes occur. As we've seen with antibiotic resistance, some of these changes are harmful to us. But there's another side to microbial mutation. It may just be what saves us from the diseases that are slowly outwitting our medical arsenal. Kiev, Ukraine, in the former Soviet Union. Berkeley, California microbiologist Jenny Hunter-Severa has come here to find new antibiotics among some of the hardiest microbes on Earth. Ukraine's Institute of Microbiology appears unsophisticated by Western standards. Many of the methods employed here and much of the equipment are not leading edge, but the Institute's scientists are well trained in classical biology.
What this program offers is a chance to link two very different sciences, sort of the classics with the modern, and to actually have more knowledge in combining those two sciences.
Genetic engineering is beyond the capabilities of this laboratory, but there is a unique and forbidding source of genetically altered microbes nearby. Chernobyl's reactor number 4. On April 26, 1986, it exploded. A toxic plume of radioactivity spread across 100,000 square miles. It was a monumental catastrophe. At least 15,000 people have died as a result of the accident. Yuri Gleba is the director of the Ukrainian Institute and also works for an American company. He and Hunter-Severa are on their way to the reactor complex. They are mounting a unique and potentially dangerous antibiotic safari in radioactive soil.
The whole Chernobyl area is a huge petri dish. Yes, it's a huge experimental dish where you have experiments going on at a very high rate.
There's a toxic effect from radiation where organisms will either be killed instantly because they can't deal with it, or some that have evolved over time will be able to handle it, but they too will now be changed in the way they function. And that's what we're going to be looking at, are these little changes that might end up in new chemicals that could be of value to antibiotics.
Reactor number 4. Thousands of workers encased it in a 12-story high concrete tomb. It has already begun to crack.
This is where so-called red forest was. pine trees that have been killed because there was a flash of radioactivity 12 years ago. Now it's regrowing. Yeah, this is very unusual. Look at these needles here. They're much longer than normal. There were some physiological stresses that this plant went through, and maybe more than that, maybe some mutations that are causing this.
Having been exposed to radiation, which causes mutations, microbes in the soil here may have changed, and consequently may be producing new antibiotics. The Chernobyl samples now go to the Institute of Microbiology in Kyiv, where the bacterial strains they contain will be cultured. Even though most bacteria don't adapt well to life in test tubes, the Ukrainian scientists have successfully cultured more than 8,000 species from around the world. Elena Kiprianova heads a research team here.
These strains were isolated from the samples which were taken in Chernobyl reactor. And these bacteria are very interesting. Are they the same as the microorganisms which live in Kyiv or in Ukraine, or they are some different because of the radiation? Did the radiation change their properties? Maybe there are some more biologically active microorganisms than the common bacteria which are living in the Ukrainian soils. Do you understand me?
Using traditional techniques, Chernobyl bacteria are cultivated in test tubes. The chemicals they leave behind in their nutrient solutions will be screened for new antibiotics in the United States. Had the winds blown to the south the day the Chernobyl reactor exploded, the 3 million citizens of Kyiv, 80 miles away, might have had to abandon their city. They were lucky, but the tragedy continues to haunt this struggling country. Will the Chernobyl antibiotic search derive a benefit from the worst nuclear accident in history? Perhaps. With so many species of microbes yet to be studied, we will find new antibiotics, and they'll work for a while, until the microbes change the rules of the game again. We don't have to go to a faraway, radioactively altered land to find useful microbes. We can search for them anywhere. The microbial community is everywhere. Largely unexplored, it may contain many undiscovered antibiotics. So, Julian Davies searches close to home.
Oh, look, I found a feather. I can stick it in my hat, right? Yeah, sure. Yeah. But,
like a grace possible. Take a soil sample.
Oh, by that tree? Oh, yeah. Let's have a look there. Right. So here I am, essentially in downtown Vancouver, Stanley Park, the center of Vancouver. This is not an exotic environment, although some people might think so. But here is some soil. This is a bag of soil we collected just down the road, right by an old Douglas fir stump that may have been cut down many years ago. The stump is old, but the soil is typical of that particular stump. That's what's interesting. And if we go somewhere else, next to another kind of tree, we find another kind of soil. And why is it another kind of soil? Because the microbial population is totally different. And we would expect them to generate different antibiotics, a variety of different products.
Most of the microbes in Davies' soil samples can't be grown in the lab. Separated from their tiny communities, they die.
In a handful of soil, there are maybe as many as 5,000 to 10,000 different microbial species, and they live there as a community. They don't grow singly like they do in the lab. They're not growing on little petri plates with nutrients. They need the community in order to grow. In fact, 99.9% of the microbes from the environment cannot be grown in the lab. So the question is, how do we get at them?
Davies is using a new approach to find antibiotics from microbes that won't grow in the lab. He heads the University of British Columbia's microbiology department and is also the president of a new company on campus called Terrigen. Davies and his colleagues are removing the microbe's DNA, the genetic codes that contain all the instructions for the microorganisms' internal processes.
We take the genetic material that we isolate from a soil or some non-cultivatable source of microbes, and we take that genetic information and we introduce it into what we call surrogate hosts. We introduce that information into other microbes that we can grow. We are manipulating DNA. We're taking DNA from one organism and we're putting it into another organism. And we're asking that the second organism, the recipient organism, can use that DNA to provide us with novel products.
Microbes are no strangers to accepting each other's DNA. In nature, they do it all the time.
Microbes have incredible abilities to exchange genetic information. So a microbe can be resistant to an antibiotic and transfer the ability to be resistant to that antibiotic to another microbe.
Sharing genes is a fact of life in the microbial world. Let's see that again. Tiny fragments of DNA, called plasmids, are transferred from one microbe to another. A single plasmid can pass on many different genetic traits, including antibiotic resistance. Terrigen takes small fragments of DNA, rather like plasmids, from microbes that can't be grown in the lab, and implants them in microbes that can. Genetically altered microbes are grown in cultures by a robot and screened for new traits. Small paper discs are saturated with the chemicals the microbes produce. The discs are placed in colonies of harmful bacteria. Already, it appears Terrigen has found an antibiotic.
Staphylococcus aureus is an important pathogen in hospital.
We've got this very nice zone of clearing where bacterial growth has been inhibited around this disc. So we know we've got an active antibiotic here.
The clear circle means the harmful bacteria are being killed by the antibiotic.
What's really exciting to us about this one now is that it's also showing activity against Staphylococcus aureus, one of the bacteria that's been causing a lot of problems in hospitals these days. Many of the strains that you find in hospitals are resistant to almost all drugs. We're going to carry on with the purification of this one, and we're really hoping that it might be Terrigen's first new antibiotic.
A promising start, but new antibiotics take years to reach the pharmacy shelf. A better understanding of how DNA works is crucial. For instance, sometimes mutations can affect critical processes like gene regulation.
Not every part of every DNA molecule is being transcribed at the same time. Organisms turn genes on and off to make the things they need when they need them. This is called regulation. Now, some genes are working all the time, so they're continually transcribed, kind of like the way the thermostat in this house is always working. But it wouldn't be practical for all the genes to be expressed all the time. That would just take way too much energy. Sort of like it wouldn't make sense for me to turn on the TV.
Are inappropriate. You don't give antibiotics for a cold.
And the radio.
Bottom line is that antibiotic resistance has reached crisis proportions.
The heat.
People are dying every day.
So the question is, well, we
and the fan.
We need new antibiotics.
The microbial community is still
all at the same time.
And the carbon.
It would take too much energy, overload the system, and shut it down. Oh, there we go. So, to avoid blowing any more circuit breakers, I'm only going to use the things I need when I need them. Like the wiring in this house, the DNA information is always there, but But the cell doesn't need all of that information all of the time. For example, one piece of gear at a time, I've got some videotape of a protist called euglena.
It turns from colorless in the dark to green in light because pathways to make chlorophyll for photosynthesis are turned on in the presence of light and off in its absence. Cells have two major mechanisms for regulating gene expression. Repression occurs when a repressor protein stops RNA polymerase by binding to DNA, preventing transcription. When transcription is needed again, an inducer changes the shape of the repressor so it no longer binds. In feedback inhibition, molecules make their way through an assembly line of enzymes to make products like amino acids. When there is enough end product, excess end product binds to the enzyme that catalyzes the first reaction of the pathway, changing the enzyme shape. This inhibits further synthesis of the end product. DNA is replicating most of the time.
How it's expressed controls the life of a cell.
If changes called mutations occur, the cell has a damage control system and can repair the changes before they become faulty proteins. But sometimes, changes do sneak through. Regulation exists to save the cell energy so that all the information coded for in DNA is not expressed all the time.
In the next episode, we'll see how scientists have learned to use this information to manipulate these mechanisms for everyone's benefit.
It's no secret that genetic information is passed on from generation to generation. I mean, take a look around. The passing on of DNA in plants and animals is achieved by a mixing of genetic information, which occurs as a result of reproduction. And it's easy to see the genetic traits that organisms share. Microbes pass on genetic information too. While their sex lives are quite different from ours, their DNA is nonetheless successfully regenerated. Human beings, along with other complex life forms, transmit their genes only to subsequent generations. So-called vertical gene transmission means that genetic information is passed on only through the reproductive process. Microbes do this too, but in addition, they have another extraordinary ability that is a far more powerful transmitter of genetic traits.
I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll explore the three main mechanisms for microbial genetic recombination. That's transformation, transduction, and conjugation. And we'll also learn how humans have harnessed these mechanisms and applied them for the benefit of all of us.
Unlike you and I, or any other creature you can see here at the fair, microbes can pass their genes not only to their offspring, but also laterally to other microbes of the same generation. This is known as horizontal gene transmission, and it's a trait that really sets the microbes apart from multi-celled life.
To learn more about horizontal gene transmission, I'm here at Oregon Health Sciences University to meet with Dr. Maggie So, chair of the university's Molecular Microbiology and Immunology Department.
I'm interested in how these microbes can acquire virulence traits. And many of these characteristics can be acquired, can be rapidly spread among a population of bacteria through these mechanisms. The microbes in your body might be totally harmless until they acquire these traits through horizontal transmission. And by acquiring these traits, they can suddenly become more virulent. They can acquire drug resistance, for instance. So if they cause disease later, they would be harder to treat.
So is there a disease that's a good example of this horizontal transfer process?
Well, a good example of this is diphtheria, which is a disease we don't see anymore because of childhood immunization programs. The disease is caused by a bacterium called Corynebacterium. And for the bacterium to cause disease, it has to produce a toxin, which kills the cells in your body. And the gene for the toxin is transmitted to the bacterium via a bacteriophage. And a lot of toxin genes are actually transferred to bacteria through the process of phage conversion.
Phage conversion can result from transduction. Genetic material passes from one bacterium to another via a bacterial virus called a bacteriophage. The virus injects its DNA into the bacterium. The bacterium then becomes a virus factory, replicating the viral genome, making viral coat proteins, and eventually releasing hundreds of new bacteriophage.
Now, every once in a while, during replication of the viral genome, a small piece of bacterial DNA gets inserted into the coat protein as well. And the live virus now transfers this genetic information from the recipient bacteria to a new bacterium.
In conjugation, another form of genetic transfer, a donor cell comes in contact with a recipient cell by way of a conjugation tube known as the sex pilus. When contact is made, a single strand of DNA containing the genetic information goes through the pilus from the donor cell to the recipient cell. This single strand of DNA is usually in the form of a plasmid, which is an extra chromosomal, independently replicating piece of DNA. A second strand of the genetic information quickly gets synthesized in both the donor and the recipient cell.
So ultimately, both cells now have complete copies of the genetic information that was transferred.
So what's the significance of that from the perspective of someone studying disease, such as yourself?
Well, we now know that a lot of genes coding for drug resistance, antibiotic resistance, reside on plasmids. So a single bacterium in a population harboring a conjugative plasmid containing an antibiotic resistance gene can ultimately spread this genetic trait through the entire population of bacteria, and this could happen quite quickly in the lab.
In transformation, a wide variety of bacteria have the ability to bind free-floating DNA from their immediate surroundings. This DNA may then be inserted into the chromosome and become a piece of permanent genetic information. Bacteria that acquire DNA in this manner inherit the genetic traits that are encoded in the incoming DNA.
How is it that there's free-floating DNA in their environment?
Well, many bacteria have, in addition to their growth cycle, a lytic cycle. That is, they die, their cells burst, and their DNA is released into the surroundings. And living bacteria that are in the neighborhood, if they're competent for DNA transformation, will then take up this DNA. Now, there are bacteria that are very indiscriminate in the kind of DNA that they take up, such as streptococci. And then there are other bacteria that are more particular, such as Neisseria and Haemophilus, and they'll only take up DNA from their own species.
There is another highly specialized form of DNA transfer known as transposition. Pieces of DNA called transposons hop from one genome to another, and sometimes from one region of a genome to another region of the same genome. These self-sufficient transposable elements may code for antibiotic resistance.
So let's say we have a transposable element that codes for a drug resistance gene. This transposable element hops, say, from the chromosome to a conjugated plasmid, all within the same bacterium. This bacterium now makes contact with a female cell, and the conjugated plasmid gets passed to the female cell. Well, the transposable element now ultimately gets passed to a second bacterium via conjugation.
Okay, so it's sort of a two-step process.
That's right. A transposable element can't transfer itself from bacterium to bacterium. It always needs an intermediary.
Knowledge of how bacteria transfer their genes can be used for our own benefit. For instance, not only can we transfer genes between bacteria, but similar processes are used to transfer genes into eukaryotic cells, like plants. And it turns out that some of this high-tech genetic manipulation is happening in some very unlikely places. This is the village of Chunamina in Zimbabwe, southeastern Africa. There has been no rain for many weeks. The village's crops of corn and millet have failed. The only staple left is cassava, its starchy, edible tubers safe underground, protected from the scorching sun. Victor Masona is a Zimbabwean plant biologist.
Cassava is the only standing crop. When we dug up a few tubers, I mean, it was like everybody was going for them, even before they were cooked, which means there is evidence of hunger. Everyone is hungry.
Cassava has brought Masona to this village. The plant is eaten every day by 500 million people in Africa and around the world. Westerners seldom encounter it, except in tapioca pudding. But here, it is the difference between survival and starvation. When something goes wrong with this crop, there's nothing left to fall back on. And something is wrong.
So this is the African cassava mosaic virus. You know, it's a disease that affects cassava, and you can see it by the crinkling of the leaves. You know, there's a mosaic on the leaves. All the shoots that come out have got the same disease. So you can actually spread the disease by propagating cuttings from this plant. So it can also be spread by whiteflies, sucking on this plant, and then they go and suck on to the next plant, and that way they transmit the virus from one plant to another.
Masona's collaborator is plant biologist Ian Robertson of the University of Zimbabwe.
Throughout Africa, this material is dug out of the ground. They see these symptoms, but they don't know it's the virus. So they'll break off pieces and they'll pass them to their relatives. And they'll plant them, but they'll plant them with the virus in the stems. So when the thing grows, it'll be full of virus, even fuller than this. And the difference between this plant and this plant is this one will give you at least 30 tons per hectare, and this one will only give you 6 or 5 or 4 or 2.
The villain is the tiniest of all microbes, a virus. Viruses are parasites. They enter animal and plant cells and use their victims' biological machinery to reproduce. Viruses are difficult to fight. Pesticides could help defeat this virus by eradicating its insect carrier, the white fly. But chemical treatments are too expensive and also might contaminate the water supply. Victor Masona uses another line of attack, one that will genetically alter the cassava plants.
Our work involves the genetic transformation of cassava for virus resistance. What this means is that we are trying to introduce genes into cassava. We hope that at the end of the day we will have genetically transformed cassava which is resistant to African cassava mosaic virus.
If you get the gene in, then we can produce for them Victor's kind of stuff, which is going to be virus resistant. Then we begin to solve the problem.
Masona obtained a grant to pursue his experiments at the International Laboratory for Tropical Agriculture Biotechnology in La Jolla, California. He fights one microbe, the cassava mosaic virus, with another microbe called an agrobacterium. Agrobacteria routinely pass their genes into plant cells. In a test tube, Masona mixes a solution of agrobacteria and genes from the virus. An electric current punches holes in the agrobacteria. The mosaic virus genes enter. The agrobacteria then transfer the viral genes to cassava plant cells.
You're trying to vaccinate the plant, right? So we are putting a gene from the virus into the plant, so it becomes part of the plant.
Masona gives the plant just enough of the virus so it can defend itself when the real virus attacks.
And the agrobacterium transfers the gene to the cassava cells, and then we take the cells, culture them, and grow them into plants.
After 5 months of careful nurturing, Victor has a finished product, a generation of genetically altered mosaic virus-resistant cassava plants. The next step is to field test the transgenic plants in Zimbabwe's climate, with Zimbabwe's farmers, among Zimbabwe's whitefly population. Ian Robertson dreams of the day when Masona's plants will thrive throughout Africa.
I'll go through all this procedure of multiplying in the field, and in 3 years' time we'll have 200,000 for the next batch of people who need it. Then they'll get 30 tons per hectare every year, and their cousins will get it and their friends will get it, so suddenly we'll have 30 tons a year more or less permanently.
But it's not quite that simple. Genetic engineering is a powerful new technology. Its long-term effects are hard to predict. Zimbabwe's leaders are cautious. They're drafting regulations for the testing of transgenic plants. Masona cannot bring his transformed cassava into the country until the regulations are in place.
It can be very frustrating that the plants are sitting in my lab in California. I cannot proceed with the testing and the introduction of genetically engineered cassava if the regulations that guide these sort of introductions are not in place. So the people are hungry, but there's something that could help the situation, and yet I'm not able to bring it here because I'm not allowed to do so.
Biotech holds out the promise of enormous power. We can manipulate crops, animals, and even people. That's a dangerous power. So we need decent people to make the choices.
Scientists like Masona are sometimes accused of toying dangerously with Mother Nature, of trying to play God.
I don't think I'm playing God. I think if God would even be happy about that situation, because our aims are to help alleviate the problem of disease in cassava in this case, by obtaining crops that can be used by very desperate people. And I don't think that is bad.
Masona hopes he will soon be permitted to test his cassava plants in Zimbabwe. If they prove resistant to the cassava mosaic virus, a young biologist's manipulation of the power of microbes could nourish hundreds of millions of people.
To you is that plants or crops can also suffer from diseases. My research involves trying to put things into the crop, so that, or into the plant, so that it doesn't suffer from the disease.
Perhaps Masona's attempts can only be successful if 21st century science is brought to everyone through education. When genetic transfer processes like transformation, the one he uses in his work, are better understood by all of us. Experiments with DNA transformation are also conducted regularly by Dr. So and her staff at Oregon Health Sciences University.
We do DNA transformation quite a lot in the lab, and we use the technique to genetically manipulate our bacteria, create mutations that we can follow for our studies, put in new genes that we can test for their virulence characteristics. So I can give you a demonstration.
Perfect.
I streaked out a strain of bacteria onto a plate, and these have been grown overnight. I'm going to swab these cells into these tubes and add DNA to each of these tubes and incubate bacteria with DNA and let the bacteria bind and take up the DNA. Now, the DNA contains a drug resistance gene, which has been inserted into a gene that we think might make our bacteria virulent. So we're actually mutating the virulence gene by inserting the antibiotic resistance gene right in the middle of it. The bacteria take up this DNA and insert it into their genome. They're going to be resistant to this antibiotic. These plates contain antibiotics, so bacteria that have taken up DNA will be able to grow on these drug plates. So after an hour of incubation, I've taken the bacteria and I've spread them on antibiotic medium, and then I've incubated the plates, and that's an overnight incubation. And here are some plates from yesterday's transformation experiment.
Okay.
First of all, we have the no DNA control. The bacteria were not incubated with any DNA, and so you see there's no transformation. They can't grow on drug plates. Here we have bacteria incubated with a large amount of DNA, and you can see that the plate is covered with bacterial colonies.
So they inherited a great deal of antibiotic resistance.
Well, that's right. The higher the amount of DNA, the more transformation events that take place. So a larger population of bacteria have now acquired the genetic trait. Here's a plate containing the smallest amount of DNA, and you can see that there are bacterial colonies on the plate, but there are fewer of them. We are using genetic engineering to try to understand how bacteria cause disease. In addition to that, scientists are now perfecting processes of DNA transfer to higher organisms based on these age-old bacterial genetics techniques. So in the case of farm animals, it is hoped that gene therapy can improve animal husbandry. And in humans, it is hoped that gene therapy can be the answer to a wide variety of medical problems.
For instance, we used to get the insulin used by diabetics from pigs like old Prince here. Now, pig insulin is close to human insulin, but not exact. Now, we can make human insulin by using microbes. By understanding the mechanics of microbial genetics, we can manipulate those mechanics for our own benefit, from improving health to turning the tide on world hunger. Over the 3.8 billion years they've been evolving and coping with changing environments, microbes have developed a variety of mechanisms for passing on genetic information and for survival. In the next episode, we'll see that it's a skill microbes have been honing since the dawn of life.
4.5 billion years ago, hot gases coalesced into a roiling amalgam of elements that would eventually cool and differentiate into what we now call Earth. For most of scientific history, it was believed that the first nascent signs of life did not emerge until 1 billion years ago, after the Earth had had time to form a cool crust, tepid seas, and an oxygenated atmosphere. But evidence now suggests that life emerged billions of years sooner than scientists had believed possible, under conditions once assumed to be far too hostile to support even the most primitive life. Now, scientists are looking into the primitive pedigree of a microbe to find the answer to the question that human beings have pondered across the millennia. How did life emerge and develop on Earth? Yellowstone National Park. For over a century, tourists have flocked here to observe its wildlife and its geothermal spectacles. The most salient features, of course, are the geysers. Early trappers and explorers noted how the hot water flowed over patches of brilliant yellow, orange, red, and green. Microbiologists, however, would later come to appreciate these natural carpets for more than their riotous colors.
The water in these hot springs is hot enough to boil an egg. Which, by the way, is illegal, and I brought mine from home to make a point. But what does this have to do with the subject of this unit, you're asking? Microbial evolution? Well, quite a lot, actually. Because, unlike eggs, certain microbes do not cook in the boiling waters of this hot spring. In fact, these microorganisms thrive in temperatures that would be lethal to most other forms of life. These high temperature tolerant microbes are called hyperthermophiles. The hyperthermophiles' abilities to thrive under such extreme conditions suggest to microbiologists a possible link to the earliest life on Earth, billions of years earlier than scientists had believed it could have existed. The main idea behind evolution is the fact that all new species evolve from pre-existing ones. We're going to see how microbes are helping scientists peer back through the mists of time to the very origins of all species. I'm Jim Leinfelder, and in this episode of Unseen Life on Earth, we'll go back in time and down in size to the beginnings of life.
For many of us, it's very difficult to accept that we evolved from microbes, from germs. Perhaps one reason is that we, who live on average just 70 years, have such a limited grasp of time. Time, like the 4.5 billion years the Earth has been around. But try thinking of those 4.5 billion years being sped up until they all take place in an amount of time we can comprehend. One 24-hour day. If the history of the Earth were compressed into one day, microbes would be the only form of life on Earth from before sunrise until long after sunset. When would the first human beings appear on Earth? Not until a scant few seconds before midnight. German scientist Karl Stetter is returning for the 10th time to the tiny island of Vulcano off the coast of Sicily. In ancient times, this active volcano was believed to be the gateway to Hades, land of the dead.
I love these guys, these volcanoes, and there are only two ways. Either you like them or you hate them. I like even the sulfur smell. So this is an environment which really makes me excited thinking and dreaming of the primitive Earth. It's very stimulating for me.
At the age of 58, Karl Stetter is focusing his considerable energy on a monumental task: unraveling the mystery of how life began on Earth. But Stetter is not searching for fossils. He is searching for microbes, what most of us casually dismiss as germs, but are really tiny fellow residents of the planet who inhabit a world that is even more complex and diverse than the world we can see.
When I was a little child already, my parents donated me for Christmas a little microscope, and I looked in it, I saw all these many shapes of things, of amoeba and bacteria, and so I became so excited that I must work on this. Now I'm so happy that I even can search for the deep organisms in the tree of life.
The tree of life. For thousands of years, human beings have thought of life as being divided into just two branches, one for plants and one for animals. Now, through breakthroughs in the understanding of DNA, scientists are painting a very different picture of the tree of life. Their most amazing discoveries are about the evolutionary relationship between human beings and microbes. For those of us who think of microbes as germs, it's hard to believe that tiny life forms like these belong at the beginning of our family tree. Yet, that is exactly what Karl Stetter is out to prove. How? Just as any one of us might learn more about the history of our own family by tracking down our oldest living relative. Stetter searches for the hottest spots he can find, hoping to locate the modern life forms that are most closely related to the first life on Earth. But why hot spots? From studying microbe fossils, scientists are convinced that life on Earth began before 3.5 billion years ago. But from studying rocks, they've discovered that before 3.5 billion years ago, our planet was still steaming hot and covered by oceans that regularly reached the boiling point, all of which made for a riddle. What can live in boiling water? Microbes that thrive in boiling water, first extensively studied by Karl Stetter on the island of Vulcano. These amazing microbes live in an environment very much like that of the early Earth.
Some of these microbes especially the ones which grow in these high temperature environments, they are very different from our way of living. We are, for example, we like to have roast pork, and on the other hand, we need oxygen to do our respiration. And these guys, instead of pork, they like hydrogen gas, and instead of oxygen, they like sulfur, and you see it all here, yellow. That's a stinky gas of stink bombs.
These microbes inhabit such remarkable homes as the boiling mud on the ocean floor near Volcano.
The temperature is about 90 to 95 centigrade, so it's really damn hot, this sediment.
Stetter is on a mission to gather another sample of these microbes he affectionately calls bugs. He is particularly interested in a species he named Thermatoga marittima.
A lot of high temperature bugs are already in, and this will be very interesting to look at it in the lab.
A lot has happened in the intervening billions of years. Life has evolved from simple single-celled organisms to the myriad diversity of life on Earth. Nowhere is that diversity more dazzlingly displayed than in the tropical rainforests that encircle the Earth's equatorial middle.
Rainforest environments are a nursery for species diversity, accounting for about 1/3 of all the plants and animals on Earth. But faced with all that variety, when you look at the natural world from a genetic perspective, what is perhaps even more astonishing is the profound sameness of all living creatures. By sameness, I mean all organisms contain DNA as genetic material. All organisms show the same basic steps in genetic information flow. And the biochemical machinery in biological information flow is the same in all organisms. All mammals share 97% of all genes. 3% is the small portion that differentiates species and organisms from each other. There are patterns that run through all living things. These patterns are what suggest that all living things came from the same basic blueprint.
Macrofossils such as the fearsome-looking elasmosaur or this Allosaurus behind me here in hot pursuit of a Stegosaurus can only take us so far back in Earth's history, between 140 and 180 million years ago. Now, as important as large fossils like this are to our understanding of the evolutionary process, they're few and far between and really can only provide an incomplete picture.
Microfossils, which are rare, give evidence of bacterial life that existed 3.5 billion years ago. But even this evidence won't take us back to the very origins of life.
To get a clearer picture, we need to look further back in time than fossils can take us, into the DNA, through the prism of living cells.
One way to think of DNA is as the recipe for producing a particular kind of organism, in the same way that a musical score is the recipe for producing a particular piece of music. In music, hard as it is to believe, the instructions for all the wonderful and complex songs ever written are made up of a finite number of notes. In DNA, hard as it is to believe, the instructions for all the wonderful and complex organisms ever produced, including us human beings, are made up of just 4 different chemicals. In music, this limited number of basic notes have been arranged and rearranged in an almost infinite number of combinations to produce unique scores for all of the songs ever sung. In DNA, the basic chemicals have been arranged and rearranged in an almost infinite number of combinations to produce a unique score, what scientists call a genome, for every organism that has ever existed. from bacteria to boys. In the 1960s, most scientists believed that creating a new tree of life by comparing the DNA of different species was impossible. But in Urbana, Illinois, Carl Woese, a jazz aficionado and molecular biologist, disagreed. For Woese, creating a tree that showed how life on Earth evolved from the first form of life to the last became an obsession.
I knew that this was a jigsaw puzzle that had to be put together. Now, in my case, I emphasize had to be, because I understood that to really be a biologist, to really understand biology, you had to understand where everything came from.
Woese focused first on the evolution of the first forms of life, microbes. The problem that had stumped everyone else was that when it comes to microbes, you can't tell much about how closely they're related from just looking at them. But Carl Woese had an ace in the hole, a technique that involved analyzing a unique chain of genetic material that exists inside every living thing, ribosomal RNA. Woese's technique involved looking at the essential components of the genetic code. By breaking the molecules into shorter sections, Woese used radiographic techniques to identify the individual pieces. Then, Woese began the painstaking process of decoding the nucleotides, labeling each either A, C, G, or U, and slowly revealing their sequence. Why go to such Herculean lengths? Because Woese believed that this chain of nucleotides, the blueprint for life, could show him how life on Earth evolved. He had learned that this chain has done its job so well that it has been passed down from the first forms of life to all life that followed. Woese realized that by comparing the thousands of A's, C's, G's, and U's in the ribosomal RNA chains of two different species, he would be able to see how much of the score of this special chain they had in common. A ribosome's function is to make proteins, regardless of the organism. Ribosomes equal protein synthesis. We can safely presume that ribosomes evolved very early because proteins are the building blocks of all living things. Ribosomes themselves are composed of a number of proteins and various types of RNA. What Woese discovered was that the sequence of nucleotides in one of these RNAs varies in different organisms, and that the patterns of differences reflects evolutionary change between organisms. In other words, ribosomal RNA is a document of evolutionary history. The more of the score any two species had in common, the more closely related Woese believed they had to be on the tree of life.
And it was indeed just like putting a jigsaw puzzle together. You don't know at all what the picture is, and then you put all these little islands together and you say, oh, here's a part of it.
One key to Woese's approach was to search for the places where major new branches of life began by looking for the place where a group of A's, C's, G's, and U's showed up in an order he'd never seen before. It was like only having heard notes arranged to play classical music your whole life, and then suddenly hearing them arranged in a rhythm and blues beat. And slowly but surely, one after another, the major branchings on the tree began to appear.
You could feel the thing growing. The islands became joined, being bigger islands, and then all of a sudden you see, this is what the picture is going to be about. And you gleefully throw in all the other pieces as fast as you can.
After more than 10 years, Carl Woese completed the puzzle. A new tree of life, created by comparing the ribosomal RNA as an indicator of the genetic makeup of different organisms. When it comes to the branch of the tree that happens to include us human beings, Woese's tree points to a new, very different way of looking at ourselves. The new tree is based exclusively on how similar the genetic instructions of different species are. And the fact is that, just like the scores of country songs are remarkably similar to the scores of rock and roll songs, the scores of plants are stunningly similar to the scores of animals and human beings. And so the Woese tree, far from being just two branches divided between animals and plants, has only allotted animals and plants tiny twigs together out on the end of the branch we share with all visible life.
It's difficult to see this tree here as your relative, because ever since recorded history almost, mankind has distinguished between animals and plants. These were the two great forms of life, and they were as different could be. But yet, scientists began to find out that there was a lot of commonality between plants and us, and that's the thing that has really sort of shocked even me, to find out not how different animals and plants are, but how much alike they are.
If you have a hard time believing that animals and plants are alike, consider what it takes to be different. When Woese decoded the scores of a newly discovered group of microbes, including a species that eats volcanic gases in the ocean, he found the letters arranged in a pattern far, far different than any he'd seen before. It was as if Woese had discovered a new kind of music that human beings had never heard before. Woese found the same unusual pattern in some of the microbes that Carl Stetter had collected from hot springs. Woese realized that their unusual genetic material made these strange organisms so different from the bacterial branch and from our branch that they had to be given their own branch on the tree of life, which he named the Archaea, for ancient. Woese's work was especially meaningful to Carl Stetter's work, for it shows that the genetic progenitors of all three branches of the tree lived in boiling water.
Due to the work done by Carl Woese, you can find out where these little tiny bugs are in the tree of life. And so it was so interesting, the hotter the temperature, the deeper the branches are, and this makes my work even more exciting.
Our concept of the evolutionary tree has changed over the years. By studying microbes and examining genetic material, other scientists have created new, more accurate versions of the tree of life model using whole chromosome data. This new model is rooted in the comparison of entire genomes, revealing that the genetic relationship of human beings and all the categories of life are linked to each other. Now that we have the ability to decipher the entire genetic score of an organism, we are in a position to truly understand the evolutionary relationships between all organisms. And this knowledge will allow us to comprehend the scope of the diversity of microbial life, the subject of the next episode of Unseen Life on Earth. The variety of life on Earth is enormous. And much of it we can't even see. Dr. Carl Woese, with his research that used RNA comparison, gave us a different tree of life, one that includes a different perspective on microbes. We learned there are ancient microorganisms at the base of the tree, like the boiling mud-dwelling Thermatoga. We learned that we are all related, though the diversity of microbial life far exceeds the diversity of all other species of life put together.
The diversity of microbes and the places in which they live is truly astonishing. Look in virtually any environment on Earth, even one as inhospitable as this, and you'll find microbes living there. I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll discover how microbes came to be so diverse, and how those differences are driven by their metabolic variety.
The changing environments of the planet and the corresponding microbial genetic shuffling through eons of time have led to enormous differences in cell structure, as well as even greater diversity in metabolic capacities. Each unique metabolism allows an organism to exploit the resources of a particular environment.
Though we all share the same chemical code of life, DNA, all life forms also differ genetically. Whoa, well these are new. You know, these skates are kind of like a mutation. Some organisms experience mutations that may or may not be beneficial depending on the environment. I can tell you that these skates don't exactly give me an edge in this environment. But if a mutation is selected for, that is, if the environment changes to give the mutated organism a selective advantage, like this, well then the mutation not only survives, but eventually spreads. This is the basis for the diversity of life. Different mutations in different environments give rise to different successful strategies for survival. Often, enough of the old conditions remain the same, and some members of the previous community persist. Perhaps in a more restricted scope.
So, as environments change, new niches become available, and organisms adapt to fill them. Because of the niches they inhabit, their strategies for survival, their way of getting and using energy, their metabolisms, differ. Metabolic diversity follows from genetic diversity.
But just what kind of microbial diversity are we talking about? Well, I'm here on the campus of Michigan State University to talk to Dr. Tom Schmidt, an assistant professor of microbiology who specializes in the study of microbial diversity. Dr. Schmidt, Jim Leinfelder.
Hello, Jim.
I think most of us have a general sense of what diversity means, but in the context of microbiology, what does diversity mean?
Jim, we already know about a tremendous diversity of microbes, and there are certainly many more yet to be discovered. And when we talk about microbial diversity, generally we're speaking about genetic diversity, how similar or dissimilar organisms are at the level of DNA. But we also consider metabolic diversity, how organisms make a living. If you think of animals and their energy metabolism, for instance, it's all pretty much the same. Animals eat plants or other animals and then oxidize those organic compounds in the presence of oxygen. That takes place in the mitochondria, and there's really not much variation on that theme. They all do it about the same way. But if you look in the microbial world, there are a tremendous variety of ways that organisms make a living, that is, produce energy to maintain cellular functions and to reproduce.
Now, In an abstract sense, I get what you're saying, but it's sort of hard to picture just sitting here.
Mm-hmm. Well, Jim, let's head over to the lab. We can take a look at some of the microbes and get an idea of how we determine their genetic diversity.
Perfect.
Okay. Jim, to take a look at the diversity of microorganisms, I've selected a termite gut as an example. And the reason for selecting that is in a small volume, we have representatives of all three domains of life, the archaea, the bacteria, and the eukaryotes. So here we have some footage of the microbial life that's inside of a termite gut.
Now, I'm guessing, I'm just a layperson, but I'm guessing these are protozoa, these big ones with the flagellum coming out of them?
Sure. These are the eukaryotic organisms, the most conspicuous ones in the gut. They're larger, they have a nucleus, and can be defined on those characteristics. In the background, the smaller organisms, here are some spiral-shaped organisms, some cocci and rods. Those are members of the bacteria and the archaea. And there's not nearly the amount of morphological diversity that allows one to characterize those organisms or to assess their genetic diversity.
So if, you know, bacteria and archaea are too small to categorize simply visually by looking at them under a microscope, how do you sort them out?
Right. So what we need to do is to take a look at a part of their genome, and I'll show you how we do that. We'll actually amplify a segment of the genome. This is a gene that encodes for a ribosomal RNA, and that's contained in all life forms, from bacteria to grasses to cows. And we'll actually go through and compare the sequence of those genes, and that'll be our measure of genetic diversity amongst the prokaryotes, the archaea and the bacteria.
Great. And we can do that right here in the lab?
Yeah.
Okay.
Jim, what we have here are some samples of the amplified DNA. What we're going to do here is to load some of the sample onto an agarose gel. We use the agarose just as a means to separate the DNA. So I'm loading into a preformed well here. You see the dye moving down into the well. Okay, so now that we have all the samples loaded, we'll turn on the voltage, and that'll apply a current. The DNA will move through the gel. DNA is negatively charged, and so it'll naturally move towards the positive pole.
Okay, so the current sort of spreads the DNA out for you.
Exactly.
Into a spectrum.
It pulls it based on the size of the DNA. The smaller fragments move faster through the gel than the larger fragments.
Got it.
And we'll then be able to go into the darkroom once the DNA has been stained and visualize that. The next step is to determine the actual sequence of that band of DNA. And we'll determine the sequence and actually compare side by side the sequences that are derived from different organisms.
Okay, and so the gene sequences to a microbe, sort of what my social security number is.
Exactly. It's your social security number or your fingerprint, and by looking at that sequence, we'll be able to identify the organism. We'll know if it's something that we've seen before, or if really it's one of these new organisms that nobody else has ever seen before.
That could be exciting.
To fully understand this process, we go to the Institute for Genomic Research in Rockville, Maryland, called TIGER for short. Here, scientists use 60 gene sequencing machines driven by supercomputers to unlock the secrets of DNA from multiple genes. One project currently underway at TIGER is the sequencing of the entire score, called the genome, of Thermatoga, the microbe discovered by German scientist Carl Stetter, living in boiling mud. Dr. Karen Nelson is in charge of the project. Her goal is to determine whether Thermatoga evolved well after life began, or if it is, in fact, one of the closest living relatives of the first life on Earth.
I'm driven by being able to answer these questions that we could not answer 5 or 10 years ago with the technology we had 5 or 10 years ago. And now we're sequencing entire genomes in one go, and to me that's just absolutely amazing.
Now, Nelson is part of the greatest explosion ever in our understanding of life's diversity. brought about by the high-speed sequencing of DNA. A key part of the process is attaching 4 different color dyes, one to each of the 4 different chemicals that make up DNA. It's as if the scientists were trying to decode the score of an incredibly long, complicated song.
I said, woman.
And someone came up with a clever way of attaching a different colored dye to each of the notes. When red dye is splashed on the notes, only the C notes turn red. When green is splashed on the notes, only the A notes turn green.
What will her chances be?
And so on.
Now she's alone in the club.
Then the color-coded score is loaded into a special color-sensitive machine that sends the data to a computer. Scientists simply check out in what order the colors are displayed on the computer, and they know in what sequence the notes are being played. It sounds easy, but since scientists need a lot of DNA to work with, they have to start out with many copies of the same score. And since they have to cut all of those copies into thousands of pieces short enough to manage, what the computer spits out in the end is numerous copies of the song in many short segments, completely out of order. A real cacophony. So the hard work in this technique, called shotgun sequencing, is to go over all the pieces again and again and again until you've isolated just one copy of the notes and put them in the correct order.
It takes a lot of dedication, but the completed product is really a reward to see 30,000 sequences going into one single stretch of A, G's, C's, and T's. It's really a remarkable experience.
With Thermatoga's genome sequenced, Nelson and her team began analyzing it and were amazed by what they found. Even though Thermatoga has been classified among the bacteria, it also shares many genes with another branch of life, the archaea.
Although it's a bacteria, it has something that makes it more similar to this other branch of life. And this was a great surprise to us because we didn't think that archaea would have any relationship like this with Thermatoga.
For Nelson, this was convincing evidence that Thermatoga came into being very close to the beginning of life on Earth.
It's evident that Thermatoga belongs at the base of the tree.
The theory that Thermatoga is one of the closest living relatives of the first life on Earth is supported by the fact that every species studied so far, including human beings, has some of the exact same DNA inside as Thermatoga. To see where other organisms fit on the tree of life and how their sequences compare, we can study the organized data on a computer in Dr. Schmidt's lab.
So Jim, we've looked at some of the morphological variability in the microbial world, and we've seen how we can take those microorganisms and retrieve a portion of their genome, and looked at that on the agarose gel. And now what I'd like to show you is what the actual sequence of the ribosomal genes look like. We're looking at the sequence here from a number of organisms, and what strikes you right off is that there are regions where all of the sequences share, all the organisms, all their sequences share the same bases, like over in this region here. There are other regions of the sequence where certain groups of organisms will share a sequence in common, whereas those that border these sequences above and below here, you can see vary in their sequence composition.
So as you determine these variations in the sequences, how does that then translate into how you position these organisms on the tree of life, essentially?
Right. Well, we interpret two sequences that have nearly identical sequences being closely related. That is, that not much time has passed since they've diverged from their ancestors, whereas sequences that are more divergent, more variable in positions, have had a longer time to accumulate mutations since their common ancestor. And so we look at comparisons of all these sequences and display that result in a phylogenetic tree, which we'll look at next.
Okay.
It appears that life has three primary lines of evolutionary descent. The eukarya, this includes all eukaryotic organisms, plants and animals, but also, for instance, the protist that we saw in the termite gut, and all organisms with a true nucleus. The prokaryotes, though, turn out to be split into two distinct groups, the archaea and the bacteria. The archaea are an interesting group. They contain organisms that live in some of the most extreme environments on Earth. Some of the metabolisms that we see amongst the archaea might be those that are most similar to the ancestors of life, in particularly the capacity to grow at high temperature. That falls amongst this group known as the Crenarchaeota. We see here some of the organisms that have been isolated from the deep sea hydrothermal vents. Here are the bacteria. Let's open up this part of the tree. Within the bacteria are a number of groups of organisms. One of the larger ones is the proteobacteria. This is a group of organisms named after the Greek god Proteus, who could change his shape at will. The proteobacteria are capable of immense diversity of different types of metabolism and frequently can switch from one type of metabolism to another. Within the proteobacteria, we see many of the common organisms, like Escherichia coli from the human gut.
Best known as E. coli, right?
Yep, that's it, the lab rat of microbiologists. Jim, the final group that we might take a look at are the eukaryotes.
Dear to our hearts because we're in that.
We're there.
Yeah.
Here are some common groups of eukaryotes, right? The arthropods, chordates.
That's where we are.
Some of them are medically important, is Giardia and Trichomonas. But there are all sorts of groups of protozoans and amoebae that make up the diversity of eukaryotic life.
Well, Tom, I think I have a good feel for the tree, bacteria, archaea, eukarya, but the one gap here for me is viruses. They're not on here. Why not?
Right. It's a good observation. The viruses currently are not on this phylogenetic tree, nor will they ever be. Viruses don't carry the same complement of genes that we find in other organisms, and so we're unable to construct a phylogeny that includes the rest of life and viruses.
DNA sequencing made it possible for the first time for scientists to systematically explore the invisible world that surrounds us. Now they could ask, who lives here? Who are they related to? And what are they doing? There was just one problem. Scientists could only ask those questions about the very few microbes they had managed to grow in the lab and collect DNA from. Then, Carl Woese's colleague, Norm Pace, made a breakthrough of his own. At its heart was the new technology of cloning, which made it possible for Pace to take a single piece of microbe DNA and make as many copies of it as he needed. Pace realized that he could now collect microbes from any environment and make copies of their DNA without ever having to grow them.
My feeling was that for the very first time, the door to the natural microbial world was open to us. That for the very first time, we could understand what the makeup of the microbial world is, and that is, after all, most of biology.
The first place Pace tried out the new technique was a hot spring in Yellowstone National Park. What Pace discovered staggered even him. Yellowstone's hot springs were home to not just a few otherworldly microbes, but to a stunningly complex ecosystem filled with hundreds of different species. And it isn't just hot springs that are teeming with microbes. It is every environment on Earth. Now, scientists are reading the scores of thousands of previously unknown species of microbes, ushering in a golden age in the study of evolution and of what the tree of life on Earth is really like. Scientists have been surprised not just by how many different kinds of microbes there are, but by discovering that some of those species exist in numbers that rival the number of stars in the universe.
The distribution of life on the planet is very different than we would have thought it even a decade ago. The estimates are that more than 95% of the biomass in the open ocean is microbial. The deep subsurface and the basalts of the crust of the Earth are shot through with life. Life is in ice, life is in waters of boiling temperatures.
But why does it matter that we are surrounded by these tiny living beings? Because it turns out many of them are doing things essential to the survival of life on Earth.
The microbial world is paramount in the context of maintaining the chemistry of the biosphere. The biosphere is fundamentally chemistry. That chemistry is fundamentally driven by microorganisms.
Why should we care? We can't see these things. Well, let's do a thought experiment and take these things away. Let's take all the bacteria in the world away. Do you know what's going to happen? All the life that we can see with our naked eyes, all that life is going to disappear in short order. The microbial world is the basis upon which our whole ecosphere rests. And without it, there is no multicellular life. And a humbling thought is if you do the converse and take away all the animals and plants, you will see big adjustments in the bacterial world, but it will still be there, growing happily along and evolving.
The message of the work of Carl Woese and Norm Pace that microbes play a key role on our planet could hardly be clearer. But what will happen when we can read the entire score, not just of microbes, but of human beings? How will that change our understanding of our role on the planet?
Without microbes in all their diversity, the entire balance of the planet would be in jeopardy. But then it never would have evolved as it did in the first place. And you and I, we wouldn't even be here. Microbes are the very fabric into which all other life is interwoven. And not only can environments influence the nature of microbes, but as we'll learn in our next episode, microbes can influence the nature of their environments.
Ancient Indian cultures felt spiritually linked to animals for providing them with life's bare necessities, food and clothing. Pacific Northwest Indians made totems to appease the animal spirits. A hierarchy of animals were venerated by the native peoples for behavioral traits the tribe admired. The salmon, their main source of protein, was treated with the reverence befitting a creature they believed offered itself up willingly to ensure the tribe's survival. But there are no totems to any of the microbes. And yet, for every culture that has ever existed, or ever will, it is the microbes' astonishing abilities to chemically transform the world that make life possible for all other forms of life.
Beautiful, isn't it? Crystal clear water teeming with trout. At least that's what they tell me. No bites yet. But the pristine lake you see now was a reeking quagmire of sulfurous mud and woody debris in the spring of 1980. And before that, well, it didn't even exist.
Mount St. Helens erupted with a magnitude 5.1 earthquake that released the largest landslide in recorded history. The resulting mud flows dammed up this river valley. Once the dust settled, there was a lake here where there hadn't been before.
But it was also a complex community of microbes that helped rehabilitate Coldwater Lake here. It was the microbes that processed the sudden infusion of nutrients and metals and caused suspended debris in the water to precipitate and settle to the bottom, thus paving the way for an ecosystem that'll support stocked rainbow trout.
And so it goes, the world over. Microbes transform and regulate ecosystems large and small. An ecosystem is a physical environment along with all of the organisms that live there. Over the next half hour, you'll see a variety of these environments which depend upon microbial cycling of nitrogen, carbon, sulfur, and other essential mineral nutrients. Long before there were animals or plants or fish or insects, when dinosaurs were still 1 billion years in the future, microbes transformed the Earth's atmosphere from an unbreathable mixture of noxious gases to the oxygen-based air used by all the life forms that were to follow. Today, microbes in the ocean affect the temperature of the planet. Ocean algae produce a sulfur compound, which escapes from the ocean into the atmosphere. In the atmosphere, the sulfur particles cause moisture in the air to form cloud droplets. With many droplets, we see a very white cloud. With just a few, it looks dark. The degree of whiteness affects how much of the sun's light and heat are reflected from Earth back into space, one of the things that controls the Earth's temperature. Ongoing cycles such as this sustain life on our planet, an ecosystem sometimes referred to as Biosphere 1. But what might happen if humans attempted to recreate this success in a custom-built, enclosed environment? In Arizona in the mid-1980s, scientists built a small version of Earth. A tiny ocean, streams, grasslands, even a desert, sealed by glass and steel. Eight men and women were to live here, physically isolated from the outside, for 2 years. Biosphere 2 was a complete world. There were many species of plants and animals, 7 ecological zones. A recipe for life. Their goal was to create a self-sustaining living environment. If successful, they would prove the possibility of sustaining life on other planets, such as Mars. In August of 1991, it was all ready. The 8 biospherians were about to breathe their last air from the outside world. The concept for the biosphere life support system was straightforward: total recycling. Everything needed for life, food, oxygen, materials of all kinds, would be used and reused. Waste would be recovered and recycled. Biosphere 2 would be completely self-contained, with nothing needed from the outside world. The concept is sound, as demonstrated by this unique airtight globe called an ecosphere. Inside, water, shrimp, algae, and a mix of aquatic bacteria can survive and thrive for years. Nutrients are used and reused. Waste from one species is what another needs. In an endless cycle, all the nutrients for all the creatures are provided for. Biosphere 2 set out to do the same thing on a larger scale. For months, all was well. The food consumed was grown within the biosphere. The biospherians went about their work. All ecological zones flourished. But, unexpectedly, oxygen levels began to fall. One species after another died. The birds disappeared, then the frogs. The biospherians found it increasingly hard to breathe. Urgent efforts at a remedy failed, and in desperation, oxygen was pumped in from outside. The sacred seal was broken. Work inside continued, but the major feature of the grand experiment was gone. Many explanations were proposed for the dramatic loss of oxygen. Plant growth in Biosphere 2 was less abundant than normal, so oxygen from photosynthesis was less than expected. But this explained only a fraction of the loss. The suspicion arose that a major part of the loss was due to the tiniest creatures. That Biosphere 2 had failed in large part because of microbes. The question is, why? What went wrong? We know that the oxygen levels dropped by almost a third in only a few months, causing the extinction of many species within Biosphere 2. Yet a conscious effort had been made to introduce plant, animal, and insect species that would successfully form a food web, a web of life. But microbial species were added almost inadvertently, most of them, like Earth's, virtually unknown. The microbes did what they're supposed to do. We created an imbalance by overfeeding them. Other factors probably came into play, but there is a strong suspicion that in significant part, one of humankind's most ambitious experiments was brought down, in effect, by a compost heap. Over-fertilization of the soil created a microbial feeding frenzy. Reproduction increased dramatically to billions of trillions. This impacted the oxygen cycle. One microbe hardly makes a difference, but trillions of rapidly reproducing bacteria consumed a huge amount of the oxygen within the enclosed ecosystem. Was Biosphere 2 a failure? Yes and no.
Biosphere 2 gave us an opportunity to begin developing ideas that we need to understand how our own biosphere works in detail. Can we afford not to understand what microorganisms are and what they do? Up till now, we've gotten by without understanding them, but we take for granted that Earth's biosphere will continue to hum along into the future without the sorts of significant problems that cause almost devastating trouble for Biosphere 2. Can we permit them to remain a mystery? Before we begin predicting how our own biosphere will respond to human influences, we have to find out what these organisms do. Without the microorganisms, there could be no plant life, there could be no animal life. We would disappear.
The diversity of life is manifest all along America's eastern seaboard. Thousands of miles of coastline along the Atlantic and Gulf Coast are shallow marshes and swamps. An incredibly productive nursery for sea life, insects, and birds.
But without microorganisms, this would be little more than a sterile sea of grass. Every food web originates with a producer, a plant that converts sun energy into food energy. Now, these vast seas of marsh grass aren't being grazed by animals. This grass can only be broken down by microbes. It is the microbes who set the cycling of energy and sustain the animals that thrive in this ecosystem. Though untypical, these ecosystems are among the most productive on the planet, pound for pound, rivaling a managed Iowa cornfield.
But you don't have to take my word for it. That's Professor Hans Perl of the University of North Carolina's Institute of Marine Sciences. He's an expert on the ecology of estuaries and the role microbes play in them.
I think we all accept the idea that these estuaries are great for lots of different forms of life and that it's a very rich type of an ecosystem, but it is surprising that it all kind of rests on the back of microorganisms. How does that work exactly?
Well, basically the microbes are the processors of the organic matter that's formed here. You see this vast sea of marsh grass, which has a lot of carbon in it, but it doesn't have much nitrogen and other nutritional elements in it. So the bacteria have to act as an intermediate to make all this food out here available for the rest of the food chain. And they do so by decomposing it, first of all. And as the material becomes decomposed and can break down or form what we call detritus, particulate matter in the water, the bacteria not only make the organic matter available to organisms that would consume this organic matter, but they also add nitrogen because they can fix nitrogen from the atmosphere and add it to the detritus and make a more complete diet basically for the next level in the food web, which would be larval organisms, the invertebrates that you see attached to the stalks here of the Spartina, larval fish, insects, and ultimately birds and fish that feed on them.
When you look closely at the Spartina, I've noticed that below water level it has a film on it, sort of a dirty looking film. Is that where the microbes are at work?
That's where the action starts. That film will go right up to the tidal line, and what that film represents is the oozing out of carbon compounds from the Spartina and the bacteria being attracted by that and starting the process of decomposition and converting that carbon into a more useful source. Secondly, they add nutritional value to this material out here. You have to realize that there's an awful lot of carbon out here, but very little nitrogen. And nitrogen is a limiting factor in coastal systems. In other words, it takes nitrogen to produce a full diet, essentially, or a good food source. And there are bacteria which break down, or are associated with bacteria that break down this detritus that fix nitrogen from the atmosphere So only they can bring in this source of nitrogen to supplement the carbon.
Right.
So the bacteria are sort of forming this extra function here to enhance the dietary completeness.
A microbe's definition of an ecosystem is not necessarily the same as ours. While humans and microbes exist side by side in many habitats, you might be surprised to learn that many complex microbial ecosystems exist within the bodies of living animals. For example, a cycling of nutrients similar to that in the marsh grass is at work right here inside old Bessie.
From the perspective of a microbe, she's just the delivery system that brings cellulose to a community of microorganisms residing in her stomach, or rumen.
Actually, the cow in the marsh are very different. The cow's rumen is anaerobic. The salt marsh is both aerobic and anaerobic, and the microbes are different in each. But the overall outcome is the same. Microbes process the food. Herbivores eat grass, but none of them can actually digest cellulose. For that, they rely on internal ecosystems of cellulose-loving microbes. What Bessie and her herbivoral relatives live on are microbial byproducts. Without the microorganisms working in concert within the bovine digestive tract, cattle would be unable to convert grass to protein, which means no hamburgers and no cheese to melt over them. And without the microbial cycles that take place in the marsh grass, much of the seafood caught in coastal areas would not exist. Microbial relationships impact our everyday existence, in some cases by helping to provide us with a source of food. But when the microbial equilibrium of an ecosystem is upset, the outcome can adversely affect our quality of life.
We used to dump our waste in these rivers. And as distasteful as that seems now, there was a time when that was a perfectly natural thing to do. Microbes in the river could break down the waste without disrupting the river's ecosystem. Of course, that was back in the days before we started living in such concentrated numbers.
When too much waste is released into a river, the ecosystem is overwhelmed. Microbes use oxygen to break down the material. In this river, the microbes were metabolizing so much waste, they were using up all the dissolved oxygen in the water. Fish suffocated. And a different river ecosystem was created, one without fish. Through the use of primary and secondary sewage treatment, this river has been rehabilitated. But humans continue to negatively impact ecosystems, simply through our waste product disposal practices. Our deepening understanding of the chemical conversation within and among microbes is enabling us to devise some remarkable ways to clean up after ourselves, or rather, for microbes to clean up after us. Given time and the right conditions, microbes can degrade almost anything. Microbiologist Terry Hazen is using this microbial ability to help solve the solid waste problem. In a typical garbage dump, the most common waste is organic material like household trash and paper. It degrades very slowly.
Everybody's heard the stories about being able to read newspapers that have been in a landfill for 50 years, okay? That's because there's not enough water there, there's not enough oxygen to get the biodegradation.
At this Georgia dump, Hazen and a local contractor are pumping air and water into the refuse. The result? The resident microbes thrive. They multiply rapidly. As their numbers increase, they degrade more trash faster.
It's so much better. Such a better way to deal with our waste problems. I hope this spreads exponentially.
We can utilize resources generated doing this operation to close the loop on the solid waste issue and leave that waste generated during our generation stable so that the next generation doesn't have to pick up that cost and add it to its operating cost.
Yeah, I think this is going to spread throughout the country. I am just super excited about this. What a tremendously active microbial environment this has become. It gets up over 140 degrees, 150 degrees Fahrenheit, and it's steamy. All of that is the microbes degrading so much material so fast that they're creating this heat.
Yeah, I was just looking at it. It's got a good smell too.
Yeah.
Yeah, it got a funky smell. It's got a good cook smell.
You can actually use your senses to see how well this is working because the smell is much lower than if we had fresh garbage. This has just sort of a musty odor, whereas over here, where they haven't been using this process, the smell is so overwhelming that it'll make you throw up.
With some extra air and water, microbes degrade the dump's organic waste in a fraction of the normal time.
So we're going from 30, 40 years or more, perhaps, down to just a couple of years, or maybe in some cases just a few months.
A faster cleanup also saves money. Hazen's microbial method benefits our budget, as well as the air, soil, and groundwater. By harnessing the power of the microbe, we have at our disposal remarkable tools with abilities we are only now beginning to discover. Abilities we've witnessed in nature, in ecosystems as diverse as the wilderness surrounding Mount St. Helens to the rumen of a dairy cow. Put simply, the vast majority of microbes busy themselves with browsing for food, and in the process, they recycle elements such as nitrogen, carbon, phosphorus, sulfur, and almost all metals, making them usable for other forms of life. They transform and maintain the ecosystems that all living things depend on for survival.
Well, skunked again. Ah! Oh, hope that doesn't get infected. Which reminds me, as we'll see in our next episode, in the case of a small minority of microbes, our efforts to control them has less to do with putting them to work for us than with making sure they don't cause disease if they get inside of us.
Attention all stations, this is the NTD performing a launch status check. Verify ready to resume count and go for launch. Say go, no go. OTC. Go. Go. LPS. Go. Juncture, go. SPE. SPE, go. LRD. LRD, go. SRO. SRO, go. You have a range.
What does a spacecraft, a canning factory, and a hospital have in common? At first glance, not much. But the truth is, millions of dollars are spent and thousands of people are aggressively working to make them very similar.
T-minus 10.
At least in one respect.
6.
Microbial control.
3, 2,
1.
and liftoff.
Disinfectants, antiseptics, antibiotics. All are household words these days, and all are used to do basically the same thing: control microorganisms. But just who is controlling who here?
Sterilized and sealed for my protection. And they're on special. As we've already learned, microbes are everywhere, and they're not necessarily out to get us. They're just doing what they have to in order to survive and reproduce. However, there are times when exposure to certain microorganisms does present an element of risk. It's at these times that we attempt to control them. The level of risk and the adverse conditions that can result determines the level and type of control strategy used. I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll examine some of the ways we attempt to control contamination and disease. Each of the environments we've just seen are, to a certain extent, just like this can of beans. They are all
controlled environments. NASA, for instance, aggressively controls and monitors the external and internal environments of the shuttles and other spacecrafts. This control is an attempt to prevent microbes from outer space returning to Earth, as well as preventing terrestrial organisms from hitching a ride into space. Microbial stowaways that could contaminate experiments. In other words, you don't want to send a craft to Mars only to discover life from New Jersey. In the hospital environment, various levels of control are implemented to prevent infection, from routine floor mopping to sterilization of surgical instruments. The food industry uses various methods to keep microorganisms out of foods, remove microorganisms from contaminated foods, and hinder the growth and activity of microorganisms already in foods.
But why do we want to control microorganisms? Well, there are several reasons, but basically we want to keep them from doing what they do naturally, growing on us, our food, and just about everything else we value.
Some contamination control methods, such as those used by the food industry, retard spoilage, thereby preventing economic loss and prolonging a product's shelf life. More importantly, though, contamination control is a health issue, whether it be in a store, in the hospital, on the street, or in our very own homes. It is an attempt to prevent encounters between us, microbes, or their toxins. However, in spite of our best efforts, encounters occur, and infection can result. Infection treatment is then used specifically to fight disease, and that can involve another form of microbial control, antibiotics.
In each case, a conscious aggressive attempt is being made by humans to control the level of risk presented by various microbes that we have deemed bad. How aggressive we are depends on the level of risk.
Home sweet home, a perfect place to demonstrate two methods of control, physical and chemical. Now, our homes don't need to be sterile, but this is certainly a place where a little control can prevent a lot of problems. This raw chicken could be a metropolis of living organisms. Back when it was being processed, the meat may have been contaminated by organisms that used to live in the gut of the bird. Those organisms hitched a ride on this chicken into my house. Now, those organisms weren't, and they certainly aren't now, a threat to this chicken. But if they get inside us, they can make us sick, so we cook it. Cooking kills most of the microbes we worry about. It physically controls them. Now, this head of lettuce, on the other hand, is usually fine after a brief rinse. That eliminates most of the microbes that might have hitched a ride from the field or while basking in the fluorescent glow at the market. Remember, you're not the only one who paws the lettuce heads before choosing. All right, salad. Oops. Oh yeah, I meant to do this to demonstrate a point. The chicken, once cooked, is under control, microbially speaking, and the lettuce, after a bath, was too. But I just contaminated their respective environments. By using the same knife and cutting board, I've mixed their microbes. If only I'd used a few basic physical and chemical control methods, I wouldn't be in this culinary conundrum. If I'd used a different knife and a different cutting board, the pathogens on the chicken would have stayed on the chicken, they wouldn't have contaminated the lettuce, and therefore, me. And by thoroughly washing my hands after preparing the poultry, the soap acts as a chemical control, an antimicrobial agent, carrying unwanted pathogens down the drain. To prevent contamination, care should be taken to keep environments clean and separate. How clean is clean? Well, that depends on where you are and what you're doing there. Canning is one of the oldest, most reliable ways humans have controlled microbes. The combination of intense heat and pressure before and during the canning process creates an environment that microbes can't tolerate. It sterilizes the contents of the can. Sterilization is the destruction of all forms of microbial life. The sterile can is immediately filled with food, sealed, and heat processed. This prevents microbes such as Clostridium and Salmonella from contaminating the food inside, so the food remains safe and edible for a lot longer. It's a microbially controlled environment in a can. A sterile environment which stays sterile until the seal is broken and the food is exposed to another environment.
If I eat this now, I may ingest a few dangerous microbes, no problem. But if I decide to take off for the weekend and leave this open can of soup to commune with nature,
the microbes, who by the way never seem to rest, spend the weekend multiplying. And I eat contaminated bacteria soup rather than chicken soup.
If I'm lucky, it'll just taste horrible. If I'm not, some of the organisms thriving inside could make me sick.
A hospital is an environment where clean means many different things. It demands more awareness of and attention to microbial control than our kitchen, but can't hope to be as sterile as the inside of a can.
Jim, this is the emergency department. This is where patients...
Dr. Paul Lewis is an epidemiologist at Oregon Health Sciences University in Portland.
The different thing about the hospital is that a lot of the people that spend time in the hospital are particularly susceptible to infections. Either they've had an injury that's broken their skin or their bones, they've had an operation that's opened up their body, or because of their own illness, they have a very weak immune system that has trouble fighting infections. In addition, in the hospital, patients get exposed to things that aren't always present in the rest of the environment. The microbial environment in the hospital is different because of the sick patients that are in the hospital that carry different microorganisms, and the fact that we use a lot of antibiotics in the hospital, and the microorganisms in the hospital tend to be a little bit harder to kill.
It's in places like this where microbes can gain the advantage in people and infection can occur. That's why it's so important to prevent contamination by using techniques such as disinfection, antiseptics, and sterilization.
These methods of control attempt to destroy pathogens, thereby reducing the number of organisms to a level deemed safe by public health guidelines. Disinfection is the destruction of pathogenic organisms on inanimate objects, such as a sink or a hospital bed. Antisepsis refers to that destruction on a living object, such as skin. Disinfection and antisepsis are effective, but do not destroy entire populations. Endospores may survive. To destroy all forms of microbial life requires sterilization. One method of sterilization used in a hospital is the autoclave, moist heat under pressure. Not unlike the canning process, is used to sterilize everything from surgical instruments, some intravenous solutions, and even hospital clothing and blankets. A hospital also attempts to eliminate or control the sources, routes, or agents of infection transmission.
In the infection control program, we practice what we call 4 levels of isolation. And the first level of precautions is just called standard precautions, and this applies to all patients in the hospital. And for this, we just intend that all healthcare workers wash their hands before and after any patient contact. It's to prevent the healthcare worker from spreading any potentially infectious substance to the patient, and it also prevents the patient from spreading something to the healthcare worker, and from the healthcare worker indirectly spreading that to the next patient they encounter. The next level of isolation is what we call contact precautions. We take contact precautions in situations in which patients are infected with microorganisms which can be spread either to healthcare workers or to other patients. but they can't jump and they can't do it magically. They have to be spread by direct contact. But if a healthcare worker has direct contact with a patient, they need to be wearing gloves and to be wearing a gown to prevent contamination of their hands or clothing. The next level is what we call droplet precautions, and that's because they're spread in what are called large droplets, which actually fall out of the air within a fairly small sphere around the coughing or sneezing patient. In this situation, in addition to the healthcare workers wearing the gloves and gown to prevent contamination of their hands or clothing, they also wear a mask so that when they are providing care to the patient within that 3-foot radius, they won't inhale or have contact with their mouth or nose with those large droplets. And the final category of isolation is what we call airborne precautions. And this really is for those microorganisms that can fly. And that's actually a very small number. And that includes things like measles, chickenpox, and tuberculosis. And in these situations, we use our highest degree of isolation, in which the patients are actually kept in rooms which are actually at a lower pressure than the rest of the hospital, so that air only flows into these rooms. In addition, when a healthcare worker or visitor enters that room, they wear a special mask that actually has a high efficiency filter on it that prevents them from inhaling these organisms in the air.
Surgery is one area which demands a high level of cleanliness. Cleanliness which starts well beyond the actual operating room. Near the entryway, a line on the floor clearly indicates a point past which all must be wearing surgical attire, including cap and booties. Those who enter the operating rooms must do more to assure microbial control, starting with an aggressive hand washing.
Our surgical scrub brushes are impregnated with a variety of different disinfectants, which are known to be effective at killing the bacteria that inhabit the skin. Furthermore, the surgeons wash more than just the tips of their fingers. They wash all the surfaces of their hands and wrists, and actually up to their elbows. When a surgeon enters an operating room after having completed their surgical scrub, enter without touching the door, and before touching anything in the room, they are assisted in the donning of a sterile gown, and in general most surgeons wear at least 2 pairs of sterile gloves. The degree of cleanliness of the equipment in this environment has also reached the highest level, that all the equipment is not just clean, but it's actually sterile, and the sterility is documented.
In the emergency area, the various levels of control merge. Patients from outside the hospital enter an intensive care environment instantly. The atmosphere may seem chaotic, but the infection control protocol is the same here as any other intensive care area. Gloves, masks, and other barrier precautions are worn to protect patient and staff. The trauma rooms are stocked with prepackaged sterile instruments, IV solutions, and equipment.
There's no schedule here. When it rains, it pours. Sometimes nothing will happen for 6 hours, and then 6 patients will come at once. So it's sort of like the hurry-up version of infection control here, always being ready ahead of time and cleaning everything up as soon as each case is finished.
As soon as the room has been vacated, the cleaning process begins. First, the linen is removed to be laundered. Disinfectant is then applied to the bed, the railing, anywhere contamination may have occurred. Even the sink is disinfected. The various cleaning procedures used in a hospital are fairly successful at controlling most microorganisms. But as we've learned, microbes adapt.
A few years ago, we had a new drug-resistant organism enter our hospital environment. We wanted to make sure that our cleaning and sterilization techniques were adequate. So after a room in which a patient with this drug-resistant organism had spent a few weeks, we sent our infection control team in to actually culture the surfaces. And bed rails are one of the important surfaces that patients touch a lot and can contaminate with microorganisms. So we just simply take these culture swabs and go along the entire bed rail. And we just keep going back and forth and really covering all the surface. After we've done that, we take this swab and we'd actually send it to the laboratory, but I'll just show you what we do. We'd actually go ahead and plate it on a selective media on which only this drug-resistant organism can grow. Then we take this plate with the selective media on it, send it off to the laboratory where it'd be incubated overnight. This is the plate I just used, but the result we'd be looking for would be like this. No growth of the drug-resistant organism on this selective media plate.
No news is good news.
Yeah, exactly. If it were not effective, we'd expect to see some bacterial growth. This is just an example, not from this bed rail, of what that growth would look like. Well, as you see here on this plate, there has been growth of the organisms. Each little dot there is called a colony, and bacteria grow very quickly. Each one of those last night were just a single bacteria, and overnight they grow at a very rapid rate. They double almost every 20 to 30 minutes.
And so how did things turn out?
Well, it turns out we did this for our first 10 patients that had this drug-resistant organism that spent a few weeks each in the hospital, that our disinfection techniques were effective because we failed to ever find those bacteria after the rooms had been cleaned.
Great.
Although these physical and chemical methods are very effective in controlling environmental contamination, many are far too harsh to be used on or in our bodies. One way to control microbes once they get inside us is through the use of antibiotics. We think of antibiotics as a recent invention, but microbes have been creating and using them for billions of years. Like all living things, microbes compete with one another for what they need. These tiny chemists routinely produce a wide variety of substances to vanquish their microscopic competitors. Among their chemical weapons are substances which we call antibiotics, substances that we have learned to manufacture and use in the treatment of disease. We've had great success controlling disease in two ways: prevention and treatment. Once we understood the principles of microbial control, our world drastically changed.
There were a lot more healthy, happy kids, for one thing. The probability that most of these kids will live long and happy lives is better today than at any other time in human history.
Today, people of all ages are susceptible to far fewer bacterial infections due to the use of vaccines and antibiotics. However, we must remain vigilant in our attempts to control microbes that pose a health hazard. For even though we have virtually eradicated diseases like polio, smallpox, and measles, there will always be microorganisms that can cause disease. Viruses, for instance, are still a major threat because there are very few effective antiviral agents. And although antibiotics have been successful in controlling bacterial infection, in recent years a threat has appeared on the horizon. Overuse of broad-spectrum antibiotics has created the specter of antibiotic-resistant bacteria. And although these measures have improved the overall health of many of the world's peoples, there are many more places where controls and their benefits simply do not exist. Contaminated water, lack of education, poor hygiene. They all contribute to make well-known and well-understood infectious diseases the world's biggest killers. Germaine Honquet is a physician with the international agency Doctors Without Borders. She has worked in Africa and is now working in Honduras.
In developing countries, a major problem is that the environmental, the water supply, the sanitation conditions, the level of hygiene, the level of education, the level of nutrition of the people, in general, the living conditions of the people are poor, and this is favoring the spread of those infectious diseases.
Worldwide, 50,000 people a day die of infectious diseases such as typhoid, tuberculosis, cholera, yellow fever, and malaria. These are not exotic diseases, and the strategies to control them have been known for years. The infectious diseases most problematic here are well understood. Germaine Honquet is a frontline warrior in the fight against infectious disease. The battle she wages every day could easily affect us thousands of miles away.
It's clear that infectious disease, they do not have borders, they do not respect borders. Nowadays that people are moving so much from one country to another country, every country is at risk almost at every disease.
And people are not the only things moving more often from country to country. The days of buying nothing but locally produced foods are gone. The tomato you buy today could have been picked on an entirely different continent just a few days ago. International food distribution means that much like the microorganisms that hitched a ride into my kitchen on the chicken, microbes can hitch a ride into our food supply from all over the world.
Ever since humans discovered microbes, we've struggled to control them, from controlled environments to prevent contamination and rot, to controlled environments to prevent infection. We've used techniques ranging from sterilization to antiseptics to antibiotics to tip the scale in our favor. But as we've seen, microbes, which have been evolving for 3.8 billion years, are pretty resourceful and have come up with ways to withstand some of the control methods we throw their way.
This has led to the almost frantic search for new methods of microbial control, which hopefully we will use more wisely. We not only share this planet with microbes, our lives are intertwined. For the most part, those relationships are in equilibrium, but there are a whole range of relationships between macroorganisms and microorganisms, from helpful to harmful. Those are the focus of the next few shows. What if your life, all life, depended for its very existence on creatures that outnumber us millions of times over, yet few of us have ever seen? It does. And what if one day plant, animal, and human life on Earth began to disappear, and we didn't know why? Because we didn't understand these creatures' power to control the environment. It could happen, but perhaps it won't, if we learn to understand that we are not the true masters of life on Earth. Microbes are.
I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll explore the long evolving relationship between microorganisms and multicellular organisms and the various natures of those relationships. For the most part, we and other multicellular organisms enjoy a mutually beneficial relationship with microorganisms. For example, the bacteria residing in my large intestine help me break down food and make important vitamins. And it's largely the same case with the cows here. Without microbes in their digestive tracts, ruminant herbivores, such as cows, wouldn't have the capacity to break down cellulose and convert it to the meat and dairy products you and I rely on them for. Now, I suppose cows could have evolved the capacity to produce the catalyst you need to break down cellulose. The fact is they didn't. Instead, they evolved into a relationship with microbes that can break down cellulose and turn it into usable organic acids.
At Michigan State University, there are studies focusing on the mutually beneficial relationships between microbes and multicellular organisms. Dr. John Bresnak is a microbiology professor participating in these studies.
Hi, Dr. Bresnak.
Hi, Jim.
I think I'll pass on the handshake here. I can't help but notice I want to talk to you a little bit about the relationship between microbes and old Bessie here, and I'm struck by this appliance on the side of her. What's going on here?
Well, Jim, this is called a fistula, and it is a surgically implanted porthole, in effect, that allows one to observe and sample rumen contents. Would you like to maybe get some rumen fluid?
Oh, I'd love to, but I think maybe I'll join you in putting on a lab coat here. It looks a little messy. Now, what is the point of accessing it this way when, if we're patient, it'll come to us from this end?
Well, the ruminant animal feeds on plant material, and as you know, plant material is rich in cellulose.
Right.
But the ruminant doesn't have its own enzymes, cellulases, to degrade cellulose and other plant polysaccharides. So it carries out what's called a pre-gastric fermentation, in other words, before the stomach fermentation, and that occurs in the large rumen, which is about 100 liters in capacity.
Wow.
So the ruminant animal, as you can see, is feeding on some forage here, and it chews it to small bits, which then pass down into the rumen of the animal, and then a diversity of microorganisms, a dense community of microbes, carries out the digestion or the breakdown of plant polymers like cellulose and converts them to volatile fatty acids, organic acids, which the animal can then take up from the rumen into the bloodstream and which serves as the energy source for the animal. So would you like to maybe grab some rumen fluid and then take it back to the laboratory and get an idea of what kind of microbes are in there?
I'd be honored.
What you can do is stick your arm into the rumen, and you'll see it's pretty warm, and grab a bunch of rumen contents, and then what we can do is squeeze the rumen fluid into a little Erlenmeyer flask.
Wow. What amazes me is that this doesn't seem to bother Bessie in the least. Interesting. In a sense, if we take the long view here, ruminants, cows and other herbivores who have a rumen, more or less evolved in concert with the microbes, and the microbes evolved in concert with the ruminants.
Exactly. There are certain species of microorganisms that are found virtually only in the rumen and have adapted to that habitat and are highly competitive there, and function very efficiently with the other microbes that are present.
Well, what do you say we go back to the lab and get a look at these critters?
Let's do it.
Okay.
I'll see, Jim, in the relatively short amount of time it took us to come back to the laboratory, the fermentation in the rumen fluid has continued, and the gas production, including methane and CO2, has caused a lot of the fibrous particles to float up to the top. So the fermentation has continued while we were bringing it back.
Within the rumen itself, is there an interrelationship between those microbes, the way there's an interrelationship between the cow and the microbes?
Right. Usually in anoxic habitats where an anaerobic community is carrying out the fermentation, there's not one single microbe that does the whole job by itself. It requires a community of organisms, different physiological types, each coordinating with another to carry out the entire fermentation. There's not one organism that can do it all. For example, the products of the fermentation by one group of microbes actually serves as the substrate or food for another group of microbes, which in turn give off products that yet other organisms in the food web can use. And the overall concerted interaction of these different groups is what actually helps to carry out an efficient fermentation of the plant material. So they can be thought of like working in an orchestra where each member of the community has its own particular function.
And they're, in a sense, as vital as any of the cow's organs are to the process of digestion.
Absolutely, because without them, the cow would be starving. It's the degradation of this plant material to smaller carbon compounds that actually serves up the energy source that the ruminant animal uses.
This unique interaction with microbes isn't limited to ruminants. A similar process occurs in a much smaller host, where microbes turn wood into food.
To you and me, this is a quaint and rustic old barn. But to a colony of termites, this is a delectable supply of food. But while termites are able to eat wood, alone they could no more make a meal of it than you or I could. In order to digest the wood and extract from it the nutrients they need to live,
termites have had to evolve into a mutually beneficial relationship with microbes.
Dr. Bresnak also studies the relationship that termites enjoy with communities of microbes they host in their digestive tract.
Of course, the natural question is, this is the microbiology department, so, why aren't these over in the entomology department? Why are you keeping termites?
All right. Well, I get asked that question a lot, and the reason is that we are studying the microorganisms in the digestive tract of termites to understand how they interact with each other and also how they enhance the ability of the insect to digest wood, which is very difficult to digest in the first place.
Can we get a look at these guys?
Sure. First, although most people generally think of them as pests, as destructive pests, in fact, termites are one of our most abundant terrestrial insects, and they feed on one of Earth's most abundant plant materials, namely wood. So termites, through their activities, and in turn through the activity of their gut microbes, help to degrade this material, not only to provide energy and food for the termite, but also to recycle this carbon back to the atmosphere as CO2 for photosynthetic activity, so that the carbon cycle of nature can continue.
Again, it's really the microbes.
It's really the microbes, that's right.
These examples with the cows and the termites are both illustrations of relationships known as mutualism, a sort of you scratch my back, I'll scratch yours understanding between organisms. This is the rule rather than the exception in all ecosystems, from the smallest inside a termite to the vast ecosystem of the rainforest of Costa Rica. Although these relationships have existed for eons, the extent and significance of mutualism is a fairly new revelation for many biologists. Mutualistic relationships have been known for a long time, but the enormity of the microbial influence on other parts of the world's ecology and maintenance of all life is a new scientific frontier. Every creature in the forest environment is making something, using something, transforming something. A single tree may be home to 100 species, insects, birds, plants, vines, and thousands of species of microbes. There are 5,000 species in a teaspoon of soil. Virtually all the recycling in the forest is done by microbes. Many of these microbes need and use oxygen, just as we do. And they release carbon in the form of carbon dioxide, just as we exhale. The slime mold spreading across this tree is a collection of microbes, individually microscopic, collectively visible. Fungus, responsible for much of the forest recycling, is also formed from billions of fungal cells living together. In one way or another, microbes connect every living thing in this forest, every part of what biologists call the web of life. Ignacio Chapela is a pioneer in his field of mycology. A microbiologist from the University of California at Berkeley, Chapela's professional interests overlap with those of colleague Dan Jansen.
As you walk through the forest with Dan, he is looking at it mostly from the point of view of larger organisms, insects. I'm looking at it from the point of view of microbes. And it is fantastic, very enjoyable to see how those very different views really match and complement each other.
Ignacio Chapela's specialty is fungi. Fungus, perhaps surprisingly, is a form of microbe.
Fungi are very different to other microorganisms. We know that they are much closer to animals than they are, for example, to plants and certainly to bacteria, yet they are microorganisms. Many people don't realize that mushrooms are fungi and that fungi are microorganisms. Therefore, mushrooms are microorganisms.
Fungal cells are microscopic, but when they grow together, they become visible strands. When it's time for them to reproduce, strands such as these go through a remarkable transformation.
Fungi are the supreme decomposers. They can penetrate into pieces of wood, even leaves, and break down the carbon accumulated in these materials. All life on Earth is based upon carbon. Fungi break it down and respire back into the atmosphere carbon dioxide, making it available in such a way that, again, other organisms can take it back. So in that sense, microbes in general, and particularly fungi in terrestrial ecosystems, are the supreme recyclers.
Fungi break down complex compounds and transform them into nutrients for living plants.
The fungus helps the plant capture nutrients that the plant would not be able to capture without the fungus. And the fungus gets in return sugars and other nutrients that it also needs to survive. Fungi play also very important roles that are just as important as the decomposition. They form a vast network, that is an organized network, through the soil and up plant bodies and all over the landscape that establishes real communication networks. This communication can happen through nutrient processing, nutrient shuttling from one place to the other. There are plants that establish intimate relationships with that network and are able to send nutrients from a larger plant to a smaller one that might be suffering under the canopy and help her out, help that other plant out. And in many other ways, fungi, microbes, form a network that connects the whole forest.
The fungal network increases the speed and efficiency of recycling, aiding the lush growth of the whole forest. Lurking in the foliage is a major player in the forest drama. 50 million years ago, a particular species of insect forged a unique partnership with a certain type of fungus. The leafcutter ants collect leaf fragments by the million and store them in their nest. In the nest, a fungus decomposes the leaves. The ants eat the fungus.
The fungus provides the ants with the sole source of food. The young and the adults eat solely this fungus, which they feed the leaves to. Can you see the leaves freshly brought in here? All this material is fungus that is being grown on these leaves. These ants, of course, then would never be able to survive without this fungus. The ants provide the leaves to the fungus, and the fungus gives back some food to the ants. So it's a happy contract agreement that they have, just based on nutrition.
"50 million years ago, the ancestors of these ants invented a system so efficient that it took over 30% of all the recycling of all the forest foliage."
All of a sudden you have an ant and a fungus that join forces and start doing a totally new job in the ecosystem. Life takes a totally new face and a new form and starts working differently.
Ignacio Chapella and Dan Jansen combine their expertise in two fields of biology. Together, they are gaining new knowledge of how microbes and larger forms of life are connected, and new insights into the complex workings of the web of life. Today, all across the world, all plants depend on the microbial world to feed them nutrients. In turn, terrestrial plants supply 50% of the oxygen we breathe. Marine microbes like cyanobacteria and phytoplankton produce the rest. Through all of nature, microbes govern the cycling and reuse of nutrients. We have learned that microbes are part of all living processes, from the activities of single cells to a myriad of relationships between microbes and plants and animals. The sophistication of these relationships has developed throughout evolution. We are now learning to use these complex interrelationships to address some of the problems created by human activities. The great age of machines has multiplied our powers, but a price has been paid. The federal government's 310 square mile Savannah River site in South Carolina is one of the most polluted tracts of land in the United States. People can't live here. Microbes can. They routinely adapt to contaminants that would sicken or kill us. Methanotrophs, bacteria found in soil nearly everywhere, survive by eating natural gas or methane. What if we could expand their diet? Terry Hazen is a specialist in bioremediation, the use of multiple bacterial species, including methanotrophs, to clean up contamination.
How can we take those microbes and really utilize their extreme abilities to biodegrade material and direct it towards specific pollutants that are found in the water and the soil and even in garbage?
Microbes survive by taking in nutrients and degrading them, or breaking them down, with powerful chemicals they manufacture called enzymes. At the Savannah River site, radioactive materials for America's nuclear weapons were made for 40 years. This polluted the landscape. Microbes and their enzymes may have the power to clean it up.
When the Savannah River site was first built back in the early 50s, they started a process here where they used a lot of solvents. They stored that in those tanks, and then periodically they would release it through a pipe system underground, which was quite leaky. And so a lot of the contaminants leaked out into the ground along this linear path. And then it seeped down through the soil and got into the groundwater. Now that's where it's the problem, because once that material gets into the groundwater, then potentially it contaminates drinking water wells. These chlorinated solvents have been linked to leukemia in children and things like this.
Chlorinated solvents like trichloroethylene and dichloroethylene are among the most widespread and cleanup-resistant contaminants. Hazen tried something new. He harnessed the unique powers of methanotrophs by feeding them extra methane gas.
The bugs are great because they're everywhere, these methanotrophs in particular, and by adding the natural gas, they grow up to higher densities, higher concentrations, so they degrade the contaminants. Same enzyme that they use to degrade the methane and use the methane as a food will also degrade this contaminant. In fact, it'll degrade 250 other compounds.
The microbes turn the poisonous solvent into carbon dioxide and salt. Hazen inserted long pipes underground beneath the contamination and its natural population of methanotrophs. He pumped in air laced with carefully regulated amounts of methane gas. The better fed methanotrophs population increased 10 billion times. In less than 2 years, their enzymes eliminated nearly all the contamination.
Well, what happens once we're done? Well, we just stop pumping in the methane, bacteria die back to their natural levels, and everything's back the way it was.
As we learn more about microbial appetites and how to stimulate them, we may turn microbes into our greatest allies in the war against pollution. There are millions of exchanges of nutrients and energy between hundreds of thousands of species. Every organism gets what it needs and gives what it doesn't. And in the ecosystem as a whole, the exchanges balance.
But these relationships aren't all good. Though a microbe's only interest is to grow and reproduce, sometimes that can conflict with our own interest to do the same.
In the next 2 episodes, we'll explore both the normal and potentially harmful relationships between microbes and man, and what happens when these relationships fall out of balance. The outermost layer of the human body is home to millions of microorganisms that depend on our bodies for their own survival. These various microbes live on the surface and in the crevices of our skin, as well as in our hair follicles. To defend their home and primary food source from an onslaught of outside contaminants, they create a protective web to shield us from many harmful agents. However, sometimes an invading organism can penetrate our external defenses by finding its way in through our body's natural openings: the eye, nose, mouth, and other bodily openings. On other occasions, an opportunity presents itself for a potentially harmful organism to enter the body. Then what? Well, our body defends itself, that's what. An invading organism can trigger our body's coordinated multi-level defense system, referred to as nonspecific resistance and the immune response.
The human immune response is a very complicated system. The human body has many barriers to infection that we rely upon to protect us from harmful organisms. Now, a mere shaving cut is unlikely to cause a major infection. But harmful microbes can, in fact, enter our bodies via seemingly minor cuts or through contaminated food and water. Our bodies are well prepared to defend themselves against such exposures. I'm Jim Leinfelder. In this episode, we'll explore the human immune system, which consists of a complex series of defense mechanisms that protect us from foreign invaders. Our system is armed with a passive first line of defense, as well as a more aggressive second and third line. The first line of defense consists of a number of factors: an intact skin layer, mucous membranes, body secretions, and billions of microbes, called normal flora, that naturally live all over our body's exposed surfaces.
As we've seen, a variety of microbial populations make up our own personal microbial ecosystems, filling every nook and cranny. We've evolved together, interacting like seasoned dance partners. In fact, the number of microbes traveling around with just one person actually outnumber the Earth's human population. These microbes are for the most part friendly, providing us with a level of defense against other microbes. The microbes that inhabit the human skin are mostly bacteria and fungi, which live on water and nutrients from our body fluids. Microbial communities on the skin vary like the topography of the Earth. Dry areas of the back are sparsely populated, like a desert. While under the arm, life abounds, as in a tropical forest. These microbes protect us from being colonized by harmful microbes that may pass to us from other people. There are 600 to 700 species of organisms inhabiting us that we know about. Each person's normal flora is first acquired after we leave the safe, sterile environment of our mother's womb. Though we're born sterile, from the moment of birth through the early stages of life, we receive friendly microbes from our mother and other people who care for us. Microbes pass to us from the hand, mouth, and breast. Our relationship with these microbes is reciprocal. We provide them a home and food, and they protect us and provide us with nutrients. When we leave our mother's womb, our immune system is still fairly immature and undeveloped. Our immune system immediately begins to see and sample unfamiliar microbes. In this way, our bodies begin to recognize the microbes it sees from day one and can soon distinguish between friendly and harmful microorganisms. While there are some organisms that cause disease, there's a far greater number that are a part of us from soon after we're born and stay with us throughout our lives.
The intricate protective web created by the normal flora and an intact skin surface helps to seal us in and seal out contaminants. Our normal flora even release chemicals that can destroy some potentially harmful bacteria. Although this external defense may be very effective, it's not always foolproof. Sometimes an invading bacterium or virus can find its way into the body through the eyes or mucous membranes of the nose and mouth. When a harmful organism does find its way inside our body via a puncture wound or a natural opening, our bodies aggressively defend themselves in an all-out war.
Most microorganisms that penetrate our outer defenses are simply looking for a means to feed and reproduce. Infection, disease, or the damage or destruction of any healthy human cells is an unintentional byproduct for the invader's reproductive process. But in order for any of us to remain healthy, an immune response is needed to fight back. There are a variety of ways and areas of the body where replication of an invader can occur. A virus or a bacterium can threaten our bodies once they've found a way inside. As we've learned, a viral infection is the result of a virus's reproductive process. Once it enters a living cell, the virus hijacks the cell's machinery, its proteins and energy. Through this infection, a virus can make as many as 1 million copies of itself in hours. The once healthy human cell is destroyed, and the multitudes of virus copies now need to infect other healthy cells to ensure their own survival. We've also learned that a bacterium contains its own machinery to grow and reproduce. A bacterium grows and divides into 2 complete bacteria. These bacteria continue to multiply, increasing the population exponentially. Some types of bacteria produce damaging toxins. Additional toxins can be released when the invaded cells and the invader die. Some of these toxins can poison and destroy healthy human tissue. As the number of harmful organisms and toxins continue to increase, more healthy tissue is in danger. The white cells of the immune system's second and third lines of defense perform a coordinated interactive defense effort, bent on not only destroying an invading pathogen, but also preparing for defense against any future exposures to that same organism. The second line of defense is referred to as phagocytosis. Our damaged tissues release chemicals that attract phagocytic white blood cells to the site of infection. These scavengers are roving cleanup machines that move through the body with an amoeboid motion and gobble substances that are foreign to us. Phagocytic cells, including macrophages, attack all invaders. This initial response is referred to as a non-specific response. Phagocytes immediately recognize the invading organism as non-self and mount a counterattack. Stanley Falco is a microbiologist at Stanford University who has spent his career studying the relationship between microbes and humans. He has a particular fascination with bacteria.
It doesn't have a brain, but it knows where it is very accurately by constantly measuring things like the temperature, the acidity of its environment.
One of Stan Falco's students, Denise Monak, attached a small video camera to a microscope and filmed the interaction of bacteria with a macrophage, the killer cell, in real time.
I can't remember if this is the one that goes into a tubular...
That's right. This is the mystery. No.
Right.
And when you watch this interaction, you see the bacteria coming in contact, and quickly the cell, it gobbles them up. It's almost like it's eating peanuts. I mean, it's taking them in as quickly as it can. And most bacteria that fall into the clutches of a macrophage die a horrible death.
Foreign microbes are engulfed by phagocytes, like macrophages and neutrophils, which then use enzymes and harsh chemicals to kill the invading cells. Macrophages break the microbe into small bits, called antigens. These processed antigens provide a link between the second and third lines of defense. Like a trophy from its victorious battle, the macrophage displays antigens of the destroyed invader in its outer membrane. Then, the macrophage will present the processed organism to cells called lymphocytes. If the phagocytic cells are unable to defeat the invader on their own, then our bodies will move to the next level of response, our specific immune system.
Hey!
The conflict between an invading organism and the immune system is a lot like the competition in a football game. Sometimes the best offense is a great defense. To defeat its opponent, the immune system counters with a multi-level defense. We've seen that the first line of defense tries to block the invader at the level of entry. The second, phagocytosis, is a generalized or non-specific defense response, sort of like a zone defense. Phagocytosis attempts to both neutralize invading organisms and prevent them from damaging the body.
The third line of defense is a specific defense response. Here, the macrophage, with the processed organism marking its membrane, huddles up with lymphocytes to precisely identify the type of invading organism and then plan the strategy for the next play in the game. A specific antibody response designed to destroy that particular invader. Within the huddle, many of the lymphocytes become plasma cells, while others become memory cells.
Ready? Right.
The plasma cells, secreting the specific antibody required to defeat the invading pathogen, move into position for battle. The memory cells retain the memory of the exposure to this specific antigen and then remain dormant on the sidelines, ready and waiting for any future exposures to this same harmful invader. Plasma cells have a shorter lifespan than memory cells. In a future encounter, the memory cells immediately become new plasma cells, secreting the appropriate antibodies.
Our second and third lines of defense work in tandem to identify the particular antigen, mount a specific response, and establish a memory of the event to to prepare for any future exposures to the same invader.
Beside antibodies, our immune system mobilizes killer cells that can attack virally infected cells or cancer cells. Here, the smaller T lymphocyte attacks and destroys the larger virally infected cell. And here, several T lymphocytes attack cancer cells. Antibodies and cellular immunity are both required to rid our bodies of harmful infectious agents and abnormal cells. The white cells patrol, seeking out the invaders. The virus, seen here as black dots, infects the cells that line the blood vessels. Besides antibodies, specialized immune cells called T killer cells can push through and emit strong chemicals that destroy our cell with the virus inside.
Even though our bodies have developed strategies to protect us from invaders, microorganisms have developed strategies of their own to protect themselves from our defenses. For example, everybody's favorite picnic pal, salmonella. Organisms like salmonella can get into our bodies when we ingest improperly prepared foods. I think this is all right.
So when you do your standard searches...
Stanley Falco and his graduate students are learning how some microbes have evolved the capability to outwit the human immune system. The focus of their research is Salmonella bacteria, which commonly causes diarrhea as well as typhoid fever. Salmonella, seen here reproducing at 360 times normal speed, invades human cells to get its food, killing some cells, but not killing its human host. Salmonella bacteria have learned to defeat the body's defense mechanisms. They enter our body with contaminated food or water. In the mouth, Salmonella survive competing organisms, sticky saliva, and bacteria-killing chemicals. In the stomach, they survive in an acid bath. In the small intestine, they survive detergent-like bile salts designed to rip microbes apart. They sense chemical signals and aim toward a specialized barrier of the immune system. This is Salmonella's skill. It's learned to cross the barrier. After successfully surviving an onslaught of obstacles, the Salmonella encounters the macrophage, a scavenger cell designed to destroy invaders. However, Salmonella has a unique ability that allows it to not only survive inside the macrophage and reproduce, but also to kill the macrophage and burst out to infect more cells. Salmonella bacteria is more the exception than the rule. When encountering a macrophage, most bacteria and other foreign microorganisms are destroyed.
Salmonella responds by turning on new sets of genes that change the macrophage. And the end result really is the organism grows in the macrophage and eventually kills the macrophage.
Although the macrophage may die, we normally don't. Salmonella has successfully evolved to infect us without killing everyone it infects, ensuring its survival.
This interaction, usually it's a draw. The organism got food, it made more of itself and passed on to a new person to make more of itself. And we're happy because we now recognize this organism and will be immune to it. We've really just begun to understand the real kinds of complexities that exist in this life that we have with these organisms that inhabit us.
Based on what we've learned about human defense mechanisms, the medical community can now provide patients with acquired immunity to certain microorganisms in the form of vaccinations.
By introducing a vaccine, we are educating our immune system to respond as though it has seen a particular pathogen before. In effect, developing memory cells in the third line of defense to immediately respond with the appropriate white cells and antibodies.
When someone is infected with a germ, a bacteria, or a virus, a normal consequence of that is that the body's immune system produces antibodies to fight off that infection. So that if you're re-exposed to that germ, to that virus or bacteria, you won't get sick the second time. A vaccine basically circumvents that first need to become sick by exposing the body's immune system to an antigen, to a part of that germ or that virus, allowing the body to make antibodies and having those antibodies in place to prevent infection in the future. We are on the verge right now of eliminating polio from the world the way that smallpox was eliminated in 1976. There are a small number of countries in Southern Asia and in Africa where polio is still endemic, still occurs. It's been eliminated from the rest of the world, and people are predicting that by the year 2000, 2001, polio will be eradicated from the world.
Today, it's increasingly difficult to get patients to accept vaccination. This is a growing problem that is ironically a direct result of the outstanding success that we've had with our immunization programs. Many diseases have disappeared due to effective vaccines, and most people today have no memory that measles, diphtheria, and whooping cough are devastating killers.
If you walked up to a mother or father in the 1920s or 1930s and talked to them about a way that their child could not develop diphtheria or measles or mumps or polio, they would literally beg you for whatever it was that could prevent that from happening, because people from that time period recognized that these diseases that we now think of as anachronisms and things that are maybe not that bad were serious and deadly killers of children. There's basically three approaches to making vaccines. The first is to attenuate, that is to make less virulent, a virus or bacteria. Put that into a person, doesn't cause disease, but causes the body to make antibodies. The second is to take a virus or bacteria and kill it, and inject that killed particle into the body and cause the body to make antibodies against it. The third, maybe more sophisticated, is to figure out what it is on the outer coating, what proteins or sugars it is on the outer coating of the bacteria or virus that are causing the body to make antibodies. To isolate that, to purify it, or to manufacture it de novo, and just inject that single protein or sugar into the body and have the body make antibodies against it. There are, I think, a couple of reasons why infectious disease is more prevalent in developing countries. One is because vaccines may not be available to those folks. A second is because some of the public health standards that we take for granted in this country, clean water, safe food, are not the case. The reality is that vaccine-preventable diseases are still common in countries outside the United States, and particularly in developing countries. Although we live in a community, in a state, in the United States perhaps, we actually truly live in a global village, and that the way to prevent disease ultimately is not only to prevent that disease in our community, but to prevent it around the world.
We are destined to live in a contaminated world. We are not going to live in a sterile environment. The evolution of our interaction between microbes and humans is in a constant state of flux. And so I think that we will, as we have in the past, constantly be surprised by new kinds of interactions that we will view as disease and the microbes will view as survival.
Our relationship with microbes is a constant balancing act. Our bodies continually fend off the advances of ever-evolving microbes. We'll never eliminate them, so there's a constant give and take. In the next episode, we'll see what happens when that balance tips in favor of certain microbes, and the result is disease.
Human beings are teeming with microbes. There are microbes on our skin, in our pores, in our mouths, on our hair, under our nails, not to mention the microbes that squirm around inside our intestines. Really, we're perfect mobile homes for them, each of us carrying our own unique combination of microbes. For the most part, we live in harmony with them. But what happens when the normal balance of microbes gets out of whack?
Disease, that's what sorry about that. I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll explore the relationship between microbes and human disease.
Infectious diseases are as wide-ranging as the microbes that cause them. We come in contact with pathogens every hour of the day. Some pathogens even take up residence in our body, and we don't even know it until we feel sick. Our own microbial flora may become unbalanced. In this case, good bugs go bad, like a yeast infection. Or we encounter known pathogens like salmonella by eating contaminated foods, or by boldly going where no human has gone before. We encounter diseases that are new to us, like Ebola.
Diseases can spread from one person to another if the environment is right for transmission. Sometimes this can be as simple as a handshake.
Some microbes, such as the ones that live in our digestive tract, are beneficial to our well-being. Other microbes, however, have been our enemies for thousands of years. Throughout history, infectious diseases have wiped out huge populations. In 1918, over 20 million people died from an influenza virus that swept the globe. Eventually, we realized that each disease seemed to have a perpetrator, often with distinct attributes such as speed in replicating or agility in beating our immune cell defenses. Then we began to dramatically change the balance in our favor. First came the concept of hygiene, washing with clean water and soap. Public health has improved dramatically thanks to fresh water supplies and sanitation. The discovery of penicillin heralded the age of antibiotics.
Antibiotics, along with vaccines, were the tools of modern science that gave us an apparent edge in the competition against pathogenic microbes.
But this edge may not last forever. Microbial killers like HIV can emerge when something tips the balance in favor of a particular microbe. Other microbes, such as Ebola, the mysterious killer that surfaced in Zaire, or Legionnaire's disease, can appear when something changes in our relationship to the environment. These can be the most lethal. Around the world, few things are feared as much as outbreaks of diseases which kill swiftly and can appear anywhere, anytime. Such was the case in New Mexico. On May 14, 1993, in a rural community of the Navajo Nation near Gallup, New Mexico, an unknown microbial killer swiftly claimed the life of this athlete. his fiancée also died. Within a week, 4 more. Dr. Stuart Levy.
One of my attendings came to tell me that he had admitted a patient that night with a disease that he hadn't seen before.
Dr. Fred Koster.
We had to rule out known viral pneumonias, known bacterial sepsis, septicemic plague.
And suddenly that patient had a cardiac arrest for no explicable reason whatsoever.
The mystery illness caused flu-like symptoms and acute shortness of breath. The victims died rapidly, within 1 or 2 days.
And then seeing this kind of on not a daily basis, but once or twice a week.
Over 50% of the patients admitted died. The lab tests at the University of New Mexico Hospital searching for the cause of the disease were unsuccessful. The disease spread throughout the Southwest. A mother of 2 in Nevada, a man repairing his house in Arizona, a teenager in Colorado. The outbreak was serious enough to start a national alert and bring in the Centers for Disease Control.
We got the call from the folks in the Southwest, and they had reached a point where they knew they needed help. We didn't know what this was. It was totally, absolutely unknown. We knew people were dying. We knew we had a problem with some kind of disease that looked like an infectious disease. It had a very high mortality rate. We didn't have a clue what the actual organism was.
C.J. Peters is the director of the Emerging Pathogens Department at the Centers for Disease Control and Prevention in Atlanta.
We had 15 or 20 suspected patients, and it could have gone to 30, 40, 300. I mean, you just don't know when the curve starts going up until you can get a handle on things. We look for absolutely everything. We look for all the bacterial agents, the viral agents, mycoplasma, fungi, you name it.
Within 19 days, the CDC identified this microbe, a hantavirus, as the dangerous killer.
There are thousands and thousands of viruses, but where did this particular virus come from? How did a seemingly unknown, deadly disease spring up in the U.S. out of nowhere? Well, scientists learned from the Navajo people that this disease had plagued them twice before.
Dr. Ben Muneda, the grandson of a Navajo medicine man, was part of the CDC investigation. He uncovered an early clue.
I went around the reservation to the traditional healers that I knew, and then I asked them about the knowledge of the mystery illness, and what they told me was quite astounding. I was listening to them describing the things almost word per word, what we were piecing together in our meetings with the CDC.
The medicine men described the deer mouse as a carrier of the mysterious illness. The deer mouse and the hantavirus have been evolving together over millions of years and have come to an accommodation in which both survive. It is only recently that humans have become part of their story.
The traditional healers told me that the way that the deer mouse will take a human life is that through their urine and their droppings, and the way it passes is through your eyes or through your mouth or through your nose.
When hantavirus is inhaled, it will infect cells, allowing it to reproduce. Inside the lungs, small air sacs are surrounded by blood vessels. Within the blood vessels, our white cells patrol, looking out for invaders. Hantavirus, seen here as black dots, infects the cells that line the blood vessels. The The white cells sense the invader, push through, and emit cytokines that inactivate the virus, but also weaken the blood vessels. Plasma leaks into the air sacs. Soon, the plasma accumulates, leaving no room for oxygen.
4 years after the first case had been diagnosed in New Mexico, 180 people had died throughout the U.S. and Canada. Over 100 more had been infected.
There are many examples of transmission of a virus or microbe from animals to humans. Plague also comes from rodents. Anthrax from cattle. In each case, something allows the microbe to expand into a new host. Something in the ecosystem tips the balance.
Once it became clear that deer mice were the reservoir species for this new hantavirus, we hypothesized that what caused the '93 hantavirus outbreak, at least in the southwestern United States, was the El Nino-driven increase in plant primary productivity, which caused more food for mice, which caused more mice, which caused more people to come in contact with infected mice, and thus more hantavirus pulmonary syndrome.
We all fear the day when another new microbe emerges, becomes mobile, and potentially sweeps the globe with disease. But what if a new killer wasn't an emerging disease, but a re-emerging one?
400 years ago, in the English countryside, a mysterious respiratory disease terrorized Tudor England. It was called the English sweating sickness for the high fevers it induced. It ravaged the countryside between 1485 and 1551. The disease even changed the course of history by taking the life of young Prince Arthur Tudor. His younger brother, Henry VIII, became king, paving the way for the English Reformation. Then, the sweat disappeared entirely. For over 400 years in metropolitan London, there has not been a single documented case of the sweating sickness. It was almost forgotten. That is, until 3 medical detectives suspected a connection between the sweating sickness and hantavirus. Dr. Vanya Gant and Guy Thwaites teamed up with Mark Tavener, a 16th century historian who could translate the Old English and Latin of ancient records. The 3 of them set out to solve one of England's oldest medical mysteries. The sweating sickness so affected Tudor England that it was the subject of one of the first books written about disease. It described all the symptoms besides high fever and sweating.
The clinical descriptions, as best described, were of a very rapid onset. One would feel flu-like. You would have joint pains, headache, feel a little bit under the weather, and then a gathering shortness of breath to the point where some 24 to 48 hours later, you were dead.
I said, this sounds exactly like the Four Corners disease, the hantavirus pulmonary syndrome that had been described maybe 2-3 years beforehand in the southwestern corner of the United States.
Thwaites and Tavener set out to find a broader source of information. Their richest came from gravediggers' records and old parish registers.
Parish records of over 680 churches showed the vast effects of the frightening disease. Gravediggers' records revealed 5 outbreaks during the Middle Ages, with most victims succumbing during the hot months of July and August.
It was certainly a summer disease. It was not a disease that was associated with poor conditions of sanitation. it was scattered, it was rural, it didn't conform to any patterns of people-to-people transmission.
The accumulating evidence led Guy Thwaites to be more and more convinced that the sweating sickness was caused by some kind of hantavirus.
What we see in modern medicine is just a snapshot. We don't see anything other than that. We don't look at history in modern medicine. We don't get taught it in our medical schools. Perhaps we should be. Perhaps we ought to get more perspective on the evolution of infectious diseases. And we think that we are seeing them for the first time. But we're not seeing them for the first time. This is a cyclical thing. They reappear and appear, and they've done so throughout the whole of history.
The relationship between the English sweating sickness and hantavirus is only speculative, but over time, pathogens do evolve, sometimes developing new characteristics that make them appear to be different diseases. 8,000 miles away from London, and just as far from the southwestern United States, is Pergamino, Argentina, the breadbasket of South America. But this remote and peaceful community is linked to those parts of the globe by an unknown danger that is lurking in the summer grass. For the local residents, it was a shock to discover hanta appearing in their surroundings without warning in a deadly new form. Dr. Delia Enria is a physician and microbiologist expert on viral diseases. At the National Institute for Human Viral Illness in Pergamino, she noticed unusual symptoms in some patients. She didn't know it was a hantavirus. Through the internet, she received a CDC report about the virus.
You are a detective. You are following step by step what we call an hypothesis.
In September 1996, 3 years after the Four Corners outbreak, Dr. Enria feared that the disease had reemerged in a different form. In the quiet resort town of Bariloche, southern Argentina, 4 people became ill with flu-like symptoms. They were short of breath, and their lungs filled up with fluid. Tests sent to the CDC proved that the killer was another new strain of hantavirus. Over 50% of those initially stricken died rapidly. The outbreak caused panic. Something was happening that had not happened in the United States. People who came in contact with victims also came down with the disease. A reporter covering the story suddenly got sick and died. A doctor who had treated patients in Bariloche became sick. He traveled 1,000 miles to Buenos Aires to see a specialist. The specialist got sick. The specialist's wife got sick. All 3 died. Delia Enria suspected person-to-person transmission, something that had never occurred with any hantavirus outbreak. This would mean the virus had changed, becoming capable of jumping from human to human. If this was the case, the virus would spread easier and faster. At the hospital in Pergamino, Dr. Enria had another case.
I got a call from Dickie Enria at the Institute for Virology in Pergamino, and she wanted to talk about the issues around human-to-human transmission. Well, I figured that that was probably just a fantasy.
Facing skepticism about her person-to-person transmission hypothesis, Delia Enria was determined to find the source of the virus. Even if it had evolved to be contagious from one person to another, the starting point, the original source, still had to be a rodent.
The Argentines organized a team. A couple of us went down to work with them. We began to set up a series of hypotheses about the way these people were getting infected. And we had a long list, and we went through the list, and we were able just to draw a line through each hypothesis until we were left with person-to-person transmission. Not supposed to happen with hantaviruses.
Person-to-person transmission of the virus could have meant rapid infection of nearby Buenos Aires or beyond. A neighbor, a relative, anyone could have passed the virus. Avoiding rodents would not prevent the disease.
The virus diseases that have been able to come into humans, cause problems, and be transmissible from human to human are the diseases that we know we have to watch out for.
Yet, in the case of this hantavirus, the disease mysteriously vanished, at least for now, almost as quickly as it had appeared. Ironically, although the virus changed strategy, its mode of transmission, it was still reaching a dead end. Either the virus kills its human host, or the host kills the virus before it can be transmitted. Humans are not a good host for hantavirus. Yet.
There are several major things, in my opinion, that are changing our relationship with microbes and with pathogenic organisms of different kinds. Climate change and changing human land use patterns.
War, famine, and changes in the microbes themselves also impact our relationship with pathogens.
Now, what's happened in modern times, as shown by the hantavirus, is that human populations have gotten bigger and bigger and bigger, and we suddenly come in contact with creatures which we ordinarily didn't before. We still have not encountered many of the microorganisms that are out there in nature. And the more we encroach in the environment and the more different living things that we affect, and indeed the more species that we wipe out, every time this happens, we really affect the microbial balance in the world in ways we can't understand.
We fear what we don't know. But as we come to understand the microbial world, we begin to realize just how interconnected our existences are.
Understanding the relationship between humanity, microbes, and the environment is a scientific challenge for the coming decades. Using this understanding to fight microbial disease is not only a scientific problem, but a social and economic one as well. The dance with microbes has lasted as long as we've existed. While some are dangerous, most are not. Besides giving us disease-fighting drugs, many help us make food, give us clean water, and have generally created the environment that sustains us. We didn't know they existed until we saw them through the microscope. Now, these earliest forms of life reveal our past and point the way toward our future. We are locked in an embrace of cause and effect with the microbial world. We do something, and they change. Then we change. And so it will continue for as long as there is life.
Annenberg Media. by the National Science Foundation, America's investment in the future. By the American Society for Microbiology, and by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
For information about this and other Annenberg Media programs, call 1-800-LEARNER and visit us at www.learner.org.